Yo sin micrófono puedo hablar nomás así. Ahorita. Bueno, pues muy buen día a todos ustedes y a aquellos que nos están escuchando por YouTube. Sean bienvenidos pues a este tercer día del Symposium de Mechanotransducción. En esta ocasión, lo que nosotros, bueno, prácticamente tenemos estos dos bloques de sesiones. En la primera sesión tenemos 3 ponentes cuyo objetivo es presentarnos diferentes tecnologías que han desarrollado en sus laboratorios para el estudio de la mecánica de las células, en conjunto tanto las técnicas como las innovaciones que han desarrollado en sus proyectos. Nuestro primer ponente, el doctor Julien, no viene directamente en presencial, pero es un investigador francés. Él básicamente se dedica al estudio de las propiedades mecánicas de las células y al desarrollar toda la tecnología necesaria para cumplir con dicho objetivo. Entonces, por favor, pues démosle la bienvenida a el doctor Julien, quien nos va a dar la primera ponencia del día de hoy. Doctor Julien, si usted está listo, por favor, ya puede comenzar con su plática.
Pues sí, muchas gracias y pues yo voy a hacer el switch rápido en inglés, si no será un poco difícil para mí y para ustedes también. So I switch. Can you hear me okay? Yes, can you hear me? Or maybe raise your hand if you can hear me. I can see you. Oh, wow. Okay. So I guess, and stop me with your hands if at some point there is a problem or whatever. So, okay, thanks a lot. It's really nice to, a nice invitation, a nice opportunity. I was saying that actually I lived in Mexico when it's more than 35 years ago. I made the calculation. It's painful. But it was a super nice experience. So it's pretty cool to have something connected to Mexico, and so I will present something in my work that is really related to mechanobiology and cell mechanics, but also more specifically, I do work and develop tools based on micropipettes. And I will. So I'm sorry, I stole your logo. And last question for the audience: can you see my mouse when I move on? Yes, I see a yes. Okay, great. So I'm interested in cell mechanics, and I also do this. This is my web page, and I also do the question is I had a leisure, and that is my hobby is to also do this representation in a kind of 3D of this cartoon representation of cells. So it's connected to my work, and I want to just to make a point. I think for the audience here, it will be already not difficult to justify, but why is it interesting to measure the mechanical properties of cells? Well, to make a point here, I show these really nice movies of microcirculations, and you see how the red blood cells are actually squeezed in these small capillaries. These small capillaries are really smaller than the size of the resting red blood cells. So it's already obvious here that the cell has to be extremely deformable not to clog the capillaries. So it's a really simple way to justify that some mechanical properties of cells will regulate some cellular processes. So every 1,000 red blood cells, typically you will find one of these, you will see one of these leukocytes or immune cells. And this is, don't tell anybody, even on YouTube, but this is really my white blood cell. And it looks different, it's more active, and it's also really mechanically really different. I should also acknowledge that really Pierre-Henri Pech, who is in the room in Mexico, is really started everything here with this. It's a seminar series on immunobiophysics, and it's also a series of workshops that will go on, including in 2025. And the study is the biophysics of these immune processes from the really nanoscales, and I think you already heard a talk about this, to really the more cellular scale, and I will talk a bit about some things we did on the physics of T lymphocytes. So I will so the common points of what I will show is actually the technology, so this micropipette based techniques. And I like to say that I do some vintage stuff, because if you look at I took the liberty of going to PubMed and measure how many papers were cited per year that were just citing keywords like micropipettes, optical tweezers, and atomic force microscope, so AFM. And you already see that micropipettes kind of reached maturity plateau like 30 years ago. So which is only happening, it has been happening only the, let's say, last 10 years for AFM and optical tweezers. And you also see the relative, it's a log scale, so the relative amount of paper citing these three different techniques that are really eloquent of the fact that there's hierarchy. Basically, it seems like a lot of people use AFM, many more than optical tweezers and many more than micropipettes. However, so interestingly, it's also inverted in terms of how much this costs. Micropipettes are basically much cheaper than optical tweezers and AFM, well, relatively cheaper. And so the question is, why is that vintage? I will try to convince you that it's actually, there's a lot to do with this technique that can be quite powerful. And I will first, it will be a bit of a catalog, but I will show you two different ways to use micropipettes. One is the most historical, is the micropipette aspiration, which basically allows you to exert a known force or maybe a known stress, mechanical stress on cells by aspirating them and to measure their deformation. So that's a way to do mechanics. Usually you need to control an applied stress to an object. Here it will be the cell, and to measure the provoked or what it implies as a deformation of your system. And the second way to use pipettes is much less standard. That's something we do more, is to use the micropipettes themselves as a flexible cantilever. So you know that it's a flexible fiber, it's a glass fiber, looking in a similar way that atomic force microscope works. So you use a flexible cantilever, and by knowing the deflection of the cantilever, you know the force you're exerting on a medium. In that case, would be indenting a cell, deforming itself, cell by pressing against it something that here is a microbit. So I will go over the two different class of ways to use micropipettes, and starting by the most, again, historical one. But first, to set up micropipettes, usually what you need is first a way to observe microscopic objects. So it's usually a microscope. And if it's for biological systems, it's an inverted microscope, because simply cells are slightly more dense than water. So they would sediment at the bottom of a Petri dish. And in this Petri dish, what you can do is plunge this micropipette. So here, this is one micropipette, which is a typically 1 millimeter glass capillary that is then has been cooled with a so-called micropipette cooler by heating the glass so that it gets thinner and thinner until it ends up by being only a few micrometers wide. So here you can see the scale. This is a single white blood cell, if typically 7 microns, and it's aspirated. Here it's just held in place by this micropipette. So the micropipette is into a Petri dish. There is a culture medium. It could be whatever medium you want, but usually we have a culture medium that really, in a way, makes cells not happy, but the less unhappy as possible. And it is then filled with something that can be pure water as soon as it doesn't contact the cell. And it's filled, then it's connected to a micropipette holder. It's just a metallic tube here, then connected to a tube to a water reservoir. And in this reservoir, here it's actually a syringe, an open syringe, but it's just used as a reservoir, where there is a level of water that can be exactly at the same level as the liquid inside the Petri dish. But if I lower the height of the reservoir, there will be a hydrostatic pressure that will aspirate cells inside the micropipette. And it's, if you did a bit of physics or mechanics, the hydrostatic pressure is given by this formula, which is extremely simple and old formula of the hydrostatic mechanics. It's the pressure difference will be given by the volumic mass of the water, in that case, g, the acceleration of the gravity field, and delta h, which is really this height difference between the reservoir and the petri dish. And if you take 1 millimeter, typically of a difference in height, you get a 10 Pascal aspiration pressure. How big or small is that? It's much smaller than the atmospheric pressure, okay, but it's more relevant to compare it to typically the effective elasticity of a cell. And it can be measured as for materials, as a Young's modulus. And this is for white blood cells. It happens to be only a few tenths of Pascal. So it's exactly the good range. You have a pressure that can be small enough to gently hold the cell, but strong enough by having several millimeters to really strongly deform it. So it exactly happens to be really the good range to work with cells. We can then pull this glass tube into something that at the end is closed almost, and the trick to cut the tip of these pipettes and open them is to plunge them into another, this is a small, you plunge them into a liquid, which is really special. This is actually a glass bead that is molten because it's like a 0.5 millimeter bead that is molten because it's put on a hot wire. The glass bead is then, it's a droplet of liquid glass. You can go into the liquid glass and you can form a meniscus. And when you stop the heating, you get trapped into the solid glass and it will break where the meniscus was placed. So you can control with a reasonably high micron or a bit better resolution to have a really pipette with the desired diameter. By using these kind of techniques, there were really beautiful works, but I need to cite a name, that's Evan Evans, who did a lot of work to characterize this way the mechanics of red blood cells, vesicles, and also these white blood cells, by again applying a non-stress and measuring either the deformation itself or the rate of the deformation, because these objects are really viscoelastic. So what's important is the amount of deformation, but also how fast it deforms again under a given constant stress, the hydrostatic pressure. So this, as you were, I mean, I showed you if you look at the dates, it's decades old. It's really an old technique, but it was really, it gave really extremely interesting information on the mechanics of cells. But there's one thing maybe it's not always mentioned, is that it's a really nice handle. Micropipettes can be really subtle handles. You see here it's used to feed neutrophils, an angry neutrophils, in a beautiful movie from Volkmar Heinrich lab, where you can see the chemotactic capacity of the white blood cell to literally smell and go to chase its target here. And here I just show it. It was in the lab. I was just playing with a white blood cell from mice, and in the preparation there were some platelets left, so you can really see how you can handle a micron scale, really micron scale object. You can really, I was aspirating, just holding it, just aspirated or not. So that's just one point to say that this is not that difficult to set up, and it's really one of the most subtle handles that we can have to handle microscopic microbiological objects. And I didn't mention that DIC stands from Hydrodynamics Lab at École Polytechnique, and this is where I do my research, and it's close to Paris. So one example, quickly, briefly, but to aspirate cell, we didn't do things where a lot was done, but in the specific context of T lymphocyte, we asked the question of just when you look at, this is a scanning electron microscopy image of T cells, T lymphocytes, and you see there's a lot of fur, there's some kind of a lot of microvillosities that suggest that there is a lot of membrane that is stored in the surface here, and you don't see it when you look at an optical movie of a cell under the optical microscope. You don't see this nano scale detail. And the question was, can we quantify it without going to the cell, but by trying to unfold this membrane until it cannot unfold anymore and until it breaks. And we can monitor the rupture of a membrane by using a trick. It's propydium iodide. When it's outside of a cell, it's dark. When it enters the cell, it becomes fluorescent because it can complex with a nucleic acid. But here we have a glimpse of bright field image, and we superimpose the fluorescence image. So as soon as the membrane breaks, we have an onset of the fluorescence, and that's basically a way to measure how much we can expand the surface area of the cell before they break. And what we showed really in a nutshell is that it kind of correlates nicely with what you can naively quantify by measuring the SEM image. We did that with different T cells or lymphoblasts or quiescent or naive T cells, and also this it's a cell line called the Jurkat cell line that is really a T cell like cell line, and so basically there's a correlation between the surface expansion at the rupture and what we can quantify from SEM. Again, quite naively, but it's an estimate, but it's a good order of magnitude. Now, the next thing we ask is, it's a really microscopic view of the cell, but now if you start unfolding this membrane, the idea is that the cell would become more tense, and you would predict that it will become effectively more difficult to deform, to press on it. So that's exactly what we do in this experiment. We aspirate the cell from the left by applying an increasing pressure. And on the right, I start using the second way I said we could use micropipettes. We use this as a really flexible pipette. It looks really big here, but it's actually super flexible. And by measuring how much we deflect it, we can measure the force we apply on the cell. And we also can measure how much the cell is compressed due to the force. So we can do really mechanics and measure the effective elasticity of a cell as a function of the membrane expansion. So, and we could show that beyond a given slack of 10 to 15%, there was a really clear correlation between how much the cell is effectively stiff and how much you expanded its surface area. And so there is when you start unfolding the cell, it becomes more tense and until it breaks, but this is for a passive cell. Now, if you look at cells that do transmigrate, so here what happens is that we have a monolayer of endothelial cells, the cells that are lining the inner part of the blood vessels. And what happens sometimes in physiological situations is that you have white blood cells that go from the blood to inside the tissue by crossing this barrier. They go really through the monolayer. That's happening. It's not that easy to see, but this one was actually crossing from above to below the monolayer. If you look at this from much higher resolution, this is a bright field image with two endothelial cells. And below, there is an interference image, basically an interference contrast, basically where you can see kind of the fit of the cell that is coming from above to below this right endothelial cell. And it's just telling you that you can monitor transmigration, and this is high resolution, but we stayed at the lower resolution. And what we did is naively measure the estimated surface area of the cell before and after transmigration, which takes place in typically 2 minutes. And what we saw is that the increase in surface area was going from this value to this value, and this is so much higher than what we measure by unfolding with a micropipette aspiration. That just meant simply that the conclusion is that there must be a huge reservoir that was mobilized during transmigration that was not present at the surface of the outer part of the membrane of the cell. So there has to be, it's a logical conclusion. It has to be that there is a huge mobilization that could be through exocytosis of inner membranes, of inner surface, in a really short amount of time, in only 2 minutes. And it's, let's say it's a point to say that this single cell mechanical measurements can tell you something that is not that obvious by only looking at. I mean, the thing is that it's really showing the huge difference between what is this surface area stores and what has to be mobilized during these transmigration events, at least in the case of T cells. Okay, so this is just an example of micropipette aspiration, which is already a bit different than just measuring this was really basically to force the expansion of membrane. And I already showed you the other way to use micropipettes is that we could compress them with a flexible micropipette by using this as a flexible cantilever. So we call this profile microindentation, but I want to point out that we were really inspired by work by Kari Dainoki-Veres, where they really already used that micropipettes as something that could hold an object and also you be used as a force sensor. And here it's pretty cool experiment where they were actually doing a mechanical assay on the worm by using again these flexible pipettes. So the idea is that here you need usually a sphere at the tip of your cantilever. For mechanical reasons, it's easy to interpret a sphere indenting another sphere or another flat substrate. So to form this sphere, we can again go to it's a microforge. It's called this device where you have a microscope with this small bead that you can melt and then play with either to cut or to steal a bit of the liquid glass in order to form a glass bead. So it's called a microforge. So here you can control the size of the bead, which can be mechanically interesting because for a given force, if you compress with a smaller bead, you have for the same force a larger compressive stress. So you can make a difference between force, the effect of force, and the effect of stress. But okay, beside this parenthesis, we could so play with the geometry of the indenter. And I have, again, this inverted microscope. Now we have a motorized version where we can move the pipette holding a single cell to push it against this micro indenter. That's basically, again a micropipette with a spherical tip at the end. And what we can do here is measure how much we apply as a force and how much the cell is getting indented, so that how much it is compressed by a distance delta here. And when you get the force versus indentation curve, you can fit it. It doesn't need to be linear. If it's a sphere indenting a sphere, it's actually, you expect something non-linear. And you can extract it from it. You can extract a so-called Young's modulus. So it's really this elastic modulus, as in material science. That's expressed in Pascal, and we call it an effective one because we know the cell is actually viscoelastic, not pure elastic. So it's really, this depends actually on how fast you push on the cell. But let's say we have an effective elasticity of the cell. And again, this is, there are really few, if not only one, extremely few mechanical models of how to extract this Young's modulus that are analytical. And it's really important here because you have basically a relationship, this Hertz model, Hertz like the researcher that gave the name of Hertz, where how you measure our frequencies, is relating the force of a sphere indenting a half infinite space with the indentation that is due to the force. So this delta or D here, the force should be proportional to delta or this indentation D to the power 3/2. And the proportionality coefficient is the Young's modulus. And so it's extremely simplistic because it's assuming that the cell is half infinite, purely elastic, linear, isotropic, and everything is wrong. It's not true. Nothing is true. All of this is a crude approximation. However, if you plot the force versus indentation during a true experiment, you retrieve a nice force to indentation power 3 so 1.5 consistently, showing that there's probably physics that are still kind of correct into this really parsimonious but really simplistic model. So we stick a lot with this Hertz model, and that's really something where we can do comparisons between different situations. One example, when we started with this profile indentation with the thesis of Lionel Guillou in the lab, what we did as a kind of a proof of concept is to monitor the indentation of cells. So, okay, one remark is that those cells are endothelial cells, they are adherent, as opposed to the white blood cells I just showed. So it's just to show that we can use on adherent cells. It's not exactly what would be like. Probably the most relevant is to do atomic force microscope, where you press vertically in that case. But here the trick we use is that we use so-called microcarriers that are really more 200 to 300 beads, or large beads for us, on which you can cultivate adherent cells. And if you take one of these large beads and you look at the equatorial plane, you can see a single endothelial cells from the side. So that's what we did. We were pressing endothelial cells from the side. And what we did actually as a proof of concept that we can monitor changes of stiffness over time is that we blew with an auxiliary micropipette. We blew a drug that was depolymerizing the actin cytoskeleton of the cell. So it's we blew cytochalasin in that case. And we could monitor over time that the elastic modulus was dropping over time quite consistently. And while I hide a bit is that we actually did a bit better than purely elastic measurement. We did viscoelastic measurement of this, and we also monitor. We showed that the cell was behaving with relax. See, it's more technical, but basically it was behaving as a power law material, where you can show that there is some kind of power law exponent of the relaxation function. This alpha that is sometimes called fluidity, and we could monitor that the behavior of the cell in terms of fluidity is also altered by depolymerizing the cell cytoskeleton. But in a nutshell, we could show that we could measure mechanical properties and follow them over time with this profile microindentation. And so we use that in several situations. We use a few, we show you some of them. But basically, this was done on several types of cells, really different kind of stiffness, and also in passive situation, but also when there is action. For instance, here at the bottom, the cell is phagocytosing this bead, is really eating it, literally, while we can monitor by pushing it in the back, monitor if it's changing stiffness. And it's a spoiler, but it does change stiffness. It becomes stiffer during phagocytosis. So this was one really non-scientific but important aspect to me, is that it leads to a lot of reasons to collaborate with different labs, labs that can be more biology or not, even mammalian cells, but also people specialized in plant cells. And I will show you actually the really recent example, because it's the thesis of Jana Husseini, who is actually co-directed with Pierre-Henri Prech again in the room probably, and René Dumaitre from the same lab as Pierre-Henri in Marseille. The idea here is not to measure exactly the elasticity, but to push against against a cell that's really specific. It's a parasite. It's much stiffer than a mammalian cell that I just showed, that a red white blood cell I just showed. It's typically 1,000 to 10,000 stiffer. The idea here is to push it against until it breaks, to basically, as opposed to purely measure the elasticity of the material, to measure its properties at the rupture, how much it can handle mechanical stress until it breaks. And it looks like this mechanically. So instead of pushing it with a bead, we push with a needle, but it's a needle, again, related to a really long flexible fiber. So what we do here is measure a force exerted by this needle and over time until there is a clear here. It's clear here. There are two ruptures actually. The first time you enter the parasite and then when you get out of the parasite. And the context here biologically is to ask whether different treatments, how you kill the parasite, it could be heat combined with bleach, and depending on the order, we change the values, and the luckily, there are really clear mechanical differences. So basically, the mechanics here give you information on the biology of the system. Let me get to another example. So the point here is more here, it's a passive system. It's not about it's again a passive system. There is no action, but the question here, it changed really of the scale has changed. It's not a single cell, it's a spheroid of many cells. And the point is that here you can, of course, adapt this kind of toolbox of microindentation. You can adapt it to go much larger scale. This spheroid is typically 100 to 200 microns. You can adapt just by changing the size, hence the stiffness of your indenter, and you can again retrieve a force to indentation mechanical curve that you can fit and get an effective Young's modulus of the whole spheroid. So it's supracellular now. And it really works actually, again, with the Ross model extremely well. So we can compare depending on the biological state of the spheroid. Let me go back to a smaller scale, go back to these single cells and single white blood cells, specifically the T cells. And also, again, the context, T cells do a lot of things, but some of them, a subset of T cells are killers, are cytotoxic, they can recognize and attack cancer cells. Others are more here to scan other cells, including healthy other white blood cells that are presenting them antigens. And you probably heard about it already during this conference. Here, what's going on, it's an immunological synapse between one of these T cells and another white blood cells. And the question that was standing some years ago was, even though you see there seems to be a lot of movements within this synapse, it's not clear whether, for instance, the T cell here in color is exerting forces on the other cell. And it's a quite general fact in mechanics, if you only measure deformations, if you don't know if the medium to be deformed is actually really soft or really stiff, you absolutely have no idea whether there are really large or small forces generated. This could happen with almost zero force if the cells that are deforming are extremely soft. So again, this calls for a direct measurement of forces. And to address the question whether these T cells were exerting forces on targets, we use a model system with, in a long-standing collaboration now with Claire Ivro's group at Institut Curie, where we use T cells from healthy donors, let's say real T cells from humans, but a really model system. Instead of a true target cell, we use micro beads with antibodies that were triggering, that were engaging the T cell receptor. Just by plating cells on a Petri dish, you can see that they seem to be quite angry and wanted to catch these antibody-coated beads. So again, by looking at this, doesn't tell you anything about if there are forces that are generated or not. You need to introduce a force sensor. So we used, I want to just jump back in time. We used at that time I didn't have this profile microindentation technology. There was what I had at hand was again an invention by Evan Evans. It's called a biomembrane force probe. And the idea here, it's pretty crazy when you think about it, but the force sensor is a cell itself. It's a red blood cell. So the idea is that a red blood cell aspirated into a pipette is becoming a nice spring. The more you aspirate it, the more tense it is, and the higher the spring constant of this spring. And now on this red blood cell, you can stick strongly a microbead with then extra antibodies that can be, for instance, triggering the activation of a T cell here. So with this, we have a probe on the right that is a force sensor, and we activated T cells and we asked, okay, what's going on? Is the cell going to pull or push, to make it simple? So it started by pushing on this bead of the sensor here. So, and then it stopped and it started pulling on the bead. It was really reproducible. On the right, what you see is actually calcium flux. That is, in a way, what you expect when you start an activation. There is a large peak of intracellular calcium. The problem here is that when it pushes on the bead, you can see it's totally deflected from the axis, and you actually cannot really quantify or measure pushing forces. So this angry large protrusion generated by a T cell, we called it this elephant trunk. And we wanted actually to measure it, to quantify it, but we could not measure, so again, these pushing forces. So one first solution was to unfold these trunks and to show that they could be super long and also quite fast. They were actually growing at the velocity close to what you expect from polymerizing actin gel. And it's suggesting, a posteriori, it's the fact that it's really full of polymerizing actin. So here we could not measure the growing, but what we could do is at some point measure when the cell growth protrusion was stopping to its growth and it was retracting, and we could measure how fast it was pulling on the probe on the red blood cell. So this was here, it was first growing, pushing the probe. We could not quantify forces, but as soon as it was starting pulling on the probe, we could measure how fast it was pulling. So what we could measure, it's called the loading rate, how fast the force was increasing over time while the cell was progressively pulling on it. And what we saw is that this loading rate in piconewtons per second was higher when the stiffness of the red blood cell was higher. So another way to say it is that the cell was adapting to the mechanical properties of its environment during activation. And that's something that resonates with a lot of other observation by others, that the fact that the cell is sensing its outer stiffness, the stiffness of its outer mechanical environment. So again, this large protrusion, when you stain it, it's really a tube of actin at the early stage, meaning the first few tenths of seconds, and there is no microtubules at this early stage. And if you look at the F-actin dynamics, the polymerization of actin by using this LifeAct probe, you can see it's a real hotspot of polymerizing actin. We also showed that these protrusions are not a pure artifact because of the presence of a bead and not another cell. We also, knowing what we were looking for, now we saw it on cell-to-cell encounters. So this is not a pure bead-dependent artifact. This also is created by T cells encountering artificial here, but presenting cells and not microbeads. Again, we moved to something where we wanted to measure these pushing forces as soon as we could, not waiting until the cell was pulling. So we went back to this. When we started developing this technology of micropipette, instead of fusing the end of the pipette, now we use this really as a micropipette that could aspirate something, and this something was this activating microbeads. So here, the force sensor is a pipette that is extremely flexible, that can aspirate a microbead, and this makes a force sensor while we can now measure pushing forces and pulling forces. So let's run an experiment of this was during the thesis of Anna Savitskaya in the lab in co-direction with Kelly Rose. We will make contact between the bead and the cell and see what happens. I mean, we already saw it a bit. now, except that we can really measure the force that this protrusion is generating over time. Then it's the cell that decides it wants to stop making this protrusion grow, and it starts recollecting and start also then pulling on the bead after it pushed on it. So we went back in a way to redid that experiment, and we quantified the mechanics of this really here with scanning electron microscopy. You will see tremendously more details of this really well-defined, again, elephant trunk, or we call it the punch also. Here I only added colors, and you see how well-defined it is, like splitting this protrusion, and that's for the youngest generation here. But anyway, I just wanted, we use CD4, so the naive T cells, but we also observe things that were looking similar in cytotoxic. So these cells that really literally try to kill the bead. They don't make it, but they try. And so it seems to be quite a general thing of T cells. And I will accelerate. I don't want to be too long here, but we quantify what you could quantify. But for instance, interestingly, between the contact with the bead, meaning engaging the receptor, and the beginning of the onset of this protrusion growth, it's actually pretty well defined, of 40 plus or minus, let's say 10, 20 seconds. It's pretty well defined, and there is something quite reproducible in the process. Here, probably helped by the fact that the bead is really stimulating, but we could do, and there was some kind of nicely reproducible behavior of even for primary T cells from one donor to the other. So now we have this picture of a growing tube full of actin, and we quickly made the thought that, okay, we know that actin is usually, the mechanical properties of cells are usually attributed in large part to the actomyosin cytoskeleton. So we saw this was full of actin. We thought probably this thing should be a thick, a stiff part of the cell. But the question is how to measure it, why it is gaining activity like this. So the idea is to do some rheology of the cell, why it is doing the thing, and this is maybe something that would be really difficult with other techniques. Here, what we did is that we have a live feedback, so we can at any time, with typically 400 Hertz, what we do is we measure the position of the pipette over a line. That's basically knowing the force over time with a relatively high time resolution. And we can feed back on the position of the piezo that will adapt to it. So that as a result, what you can do is that we can impose the cell to grow its protrusion against a well-defined control force F. But now that we can do that, we can also do the same, except that instead of constant, we can do a force that is modulated in time by a small delta F, so a small sinusoidal modulation. So under a constant force, the growth is a nicely constant increase in length of this protrusion. But if you zoom in the thickness of the line, there is actually a tiny sinusoidal modulation due to the tiny sinusoidal modulation of the force. So if you have a delta F, you measure, and delta L, you're in business to do really mechanics by measuring the stiffness of this object that is growing over time. And in a nutshell, what we showed is that the cell was indeed becoming much stiffer while it is creating this protrusion. It is becoming much stiffer, but also much more viscous. It's a viscoelastic object, and both properties are increasing together. Next thing is that, okay, and we expand these observations to other type of white blood cells, T cells, but also B cells in another collaboration, and yet in another collaboration in the phagocytes. So these are neutrophil-like cells. And in each case, even though biologically it's a really different things that's happening, different cells, different things, but in each case, there is an increase of the stiffness and viscosity of the cells. During the thesis of Alexandra Zak in the lab, we really went on this phagocytosis, and these guys, they really try to eat the bead. The bead sometimes is too large for them. They are too ambitious, and it's called then a frustrated phagocytosis. But what we showed is that during this process, again, there was an increase of stiffness of these phagocytes and also this viscosity. Nowadays at the moment, Finn in the lab is we step back and we go even before any activation. We have a so-called passive cell. This is a T cell. But when you accelerate the time, 10 times typically, passive doesn't seem to be that quiet, right? There's a lot of submicronic, but there's a lot of movement at the surface of the cell, in the cortex of the cell. And what Finn is trying to show is that by poking the cell with needles is to show that actually these local movement correspond to changes in the local stiffness of the cell. So actually, this is a way to probe this active matter. There is a T cell that is really fluctuating in mechanical properties. Then the question is, why is it used for? We don't really know, but at least we want to describe that the baseline of mechanical properties of the cell is not as quiet as we can think. It's actually quite fluctuating system. I will just briefly go over things, but I mean here just to say that we have a versatile system and we can play with our exotic thing. We can ask the question of what's going on when you force a cell to form two synapses at the same time, and it's a funny system and it works. It forms two systems at the same time, and the question is, are they really the same? We will see maybe in the coming times. And an opening to really different systems with a colleague in the lab, with Gabriel Amselem. So he's interested in micro swimmers. So this is this 20 micron thing is a single micro IG that is swimming with two flagella here on its, I will call it in its front. And these flagella are beating, it's really swimming, and each time it has a small stroke, it's actually deflecting a micropipette that is holding it, aspirating. So this is a super flexible micropipette. And each time it has a stroke, it's actually deflecting by a tiny amount, but that we can pick up, and it's basically represents several piconewtons each time it beats. And the problem is that it does that at a really high frequency, at 50 hertz. So we needed to couple these observations with a high frequency, with a high-speed camera. So we can get the 50 hertz swimming with a piconewton resolution. So it's pretty fun. We can really have a nice signal over time, super well defined, and we can really ask then biological question of what's going on during the swimming. I will pass this, but it's just to brag and to say that we have really nice resolution as opposed to what's done in the past by others. But I will quickly go, and I don't think we have time to go into detail, but I just want to say a really ongoing work is that happens by chance or as a problem, that when we tried again to look at activation of T cells, but these cells were loaded with a calcium probe. I showed you one movie with calcium, but when we actually pushed a bit the intensity of the fluorescent, it was seemingly freezing the cell, totally like making them absolutely inert, and everything stopped working. And the idea is that we went back and we tried to really probe and understand better this thing. And now we get that there's a really strong relationship between phototoxic effect and the stiffness of a cell. And I will, before I will pass the pass, but I will just show you at least qualitatively something happening again, kind of live. This cell is filled with the fluorescent probe, but the probe is not exciting. So you can take a T cell like this, push it against a solid wall, and it's fine, it will spring back. It will not be that unhappy. Now in the next movie, what I will do is that when it goes into contact and it's deformed, I will call what I make a cell pancake, because I will turn on the excitation, there will be a star, and we will see what's going on mechanically. Now the light is on, exciting the fluorophore, and the cell is totally cooked. It's really not changing anymore in terms of shape. And I will really totally squeeze all the rest, but what we did is to show that when you turn on the light, we totally freeze this activating the protrusion of the cell. We reproduce this artifact. And I want to just to make one message is that we try that in several cell types, several fluorophore. And I just want to say, be careful with too much fluorescence because there is a link between creating too much phototoxicity and stiffening the cells. I will skip that. I don't want to be too long. And I want just to wrap up by saying that we still continue developing these tools based on micropipettes and in really different contexts with different collaborators. And I want to thank them, all of them. It's really thanks to this and also all the students in the lab that went through these sometimes painful experiments. But I want also, of course, to thank you for your attention. Okay. So if there is any question, I'll be happy to answer.
¿Le paso el micrófono? Para los del público que tienen preguntas. Espere un momentito porque sí tenemos preguntas del público.
Hello. Thanks, Professor, for your great lecture. Hello, can you hear me?
Yes, perfectly. Thank you.
Yeah. I just wanted to understand, I just want to ask you about the activation of the T cells which you're performing against the antibodies.
Mm-hmm.
So is it just the antibodies which you take, or you have also tried with, you know, T cell and B cell interactions?
So, well, actually, Pierre-Henri, who is in the room, is really interested in the subject because we actually have a funding to exactly work on that, what's going on mechanically between T cell and B cells. So we just did pull into contact T cells and B cells, but without a direct measurement of the mechanical properties. We just measure basically movement, relative movement, but it was really early. And what we did is present T cells. I mean, actually, Pierre did also measurement of T cells with other cells, and there are mechanical changes. But these large protrusions, we only quantify them with these microbeads, but then we only observe them without measuring the forces with these other artificial cells presenting antibodies, but there were really no B cells. So, but they were still used as platform of artificial presenting cells that were developed by Claire Ivro's lab. So B cells specifically in our hands with these pipette measurements, no, we didn't, but basically that's something that is, or something really related to that is really ongoing in a project with Pierre-Henri.
And you have taken pre-activated cells, or you have activated the T cells through this process?
No, so these were really naive cells. Well, actually, with Claire, we quantify few of them are memory, but essentially big majority are naive cells from the blood. And in other few experiments in Claire Ivro's lab, they did pre-activate them to make lymphoblasts, and they really behave in a similar way. And also Jurkat cells that this cell line is a bit in between. They also do the same, even though it's always, I think it's something quite general. They are always more picky, these cancer cells, even though they should be standardized, they are not at all. Even the primary cells behave much more reproducibly, but all of these, they do something that is close to this last protrusion in contact of this activating microbead.
Perfect. Thank you. Thank you, sir. Thank you.
Thank you. Welcome.
Thank you. I have a quick question. So I'm not sure if I missed it, but from the last section that you were saying about the stiffening of the cells with the light essentially, right? Like the photoactivation.
Yes.
So what was the mechanism? Like why did they get stiffened when you excited fluorophore?
We don't know at all. No, I'm joking, but it's exactly, it's really an ongoing work. The idea, we can, the idea is that any fluorescent, any fluorophore will trigger and generate some intracellular ROS, so reactive oxygen species. And it's known, independently of our study, that reactive oxygen species can locally cross-link their environment. So that's our hypothesis, that we will basically cross-link the cell from inside, so almost fix it, literally, create covalent bonds, but this is not proven yet. What we did is we used this dichlorofluorescein, diacrylide, but it's diacetate, so that's something basically to make it safe permanent, but this modified fluorescein is actually a ROS sensor. So the higher the ROS levels, the intracellular ROS level, the higher the fluorescence. And we run experiments where we push the cell repeatedly, and at each time in between each compression, we shined a nice aggressive light to actually excite something which should be quite nice. It's a cell tracker red. This is supposed to be to track the cytoplasm. It's a commercial and everything. It should be nice, but it's not at all, because if you shine enough light and you just monitor using this ROS sensor, you realize that there is a nice correlation between the increase of ROS inside the cell and the increase of stiffness. So that suggests, that's not a full proof, it goes the direction that if you create intracellular ROS, you will not cook, but stiffen your cell. And we are still, we are working on a manuscript, but then there is still a lot to do to exactly understand what happens, how to what extent there is a dose dependence. There is a dose dependence. And so that's our line of thought. But, for instance, I would like to understand if I could easily prove and detect the generation of local covalent bonds. But this is a bit out of my field of expertise. And I'm not sure exactly how to attack these molecular aspects of the problem.
Thank you so much. That's pretty cool work, and I'm looking forward to reading that manuscript once published.
Thanks a lot. Thanks.
Julien, thanks for being there with us, first. And second, maybe for the audience, you could comment a bit on the effects of temperature, the way it plays on the experiments you do, and the kind of variation it will implicate.
Yeah, so first, we are pretty bad at having a nice control temperature, because when you look at, we use a high magnification, so 100x magnification. So 1 pixel is like 65 nanometer. So any drift, as soon as you have, for instance, you try to hit your microscope plate, for instance, actually it's terrible. There is a large time-dependent thermal drift. So that basically everything starts being shaky, getting out of focus. And with 100x, out of focus is pretty fast. So we try to avoid any temperature control, and we work at room temperature. So still, we did some controls with as good as we could. And when you activate T cells, for instance, at 37 versus 22-ish degrees, first, there is a kinetic effect, so things go faster. The cells start faster to grow their protrusion. Then, in terms of mechanics, I don't remember if they were becoming slightly stiffer or not during activation as opposed to room temperature, but I think, again, the kinetics was a bit faster. They were becoming stiffer a bit faster. And it's, the things that we are reassured that we don't see absolutely qualitatively different things, but there are definitely some kind of kinetic effects. So I don't know if this is one aspect you're thinking of in terms of temperature effect. But this is one at least we quantify. Another option is to have your whole microscope even table at 37. And even there, I think it's not that easy to have a drift-free situation. I think it's a difficulty you have to live with.
Y una última pregunta, que es más general para los estudiantes.
So I couldn't hear you. Yeah.
Un momentito.
Sí.
Ok, la voy a hacer en español y
Okay.
si es necesario la hago en inglés.
Vale, vamos.
Como físico, ¿cuáles son los principales desafíos que tú has identificado que vinculan el estudio de las fuerzas con los procesos biológicos?
Estoy pensando si hago la respuesta en español, pero será demasiado doloroso para mí y ustedes. O sea que otra vez voy a hacer el switch. No, lo tengo que decir. He vivido en México, he aprendido mexicano en un liceo franco-mexicano, pero soy de madre española. Y al final no sé lo que hablo, pero debe de haber un acento francés con algo que suena mexicano, debe estar un poco especial. Pero bueno, hago el switch. Then challenges. Okay, so the thing is that there is a big challenge associated with the scale. If you go to, as soon as you go to micron scale, things become difficult to handle. You know, to how do you hold a cell to inspect it is a challenge for any scientist in the world because you don't have a hand. So that's why you have this micropipette. People use optical tweezers. People try to optimize this in microchips. So this is a huge challenge. And my way to solve it is to use these micropipettes, not because I invented. I was introduced to micropipettes during my PhD, but I then continued developing and pushing it to the extreme. And so it's a challenge, which is also intellectually extremely exciting to push to the maximum the thing you have at hand, with constraints that are really difficulties, technical difficulties, but also financial difficulties. It's sometimes you, at the beginning, now I can say I can have more facility with that, but at the beginning, I didn't have the money to have an AFM, so I had to push to the limit what I had at hand, and my what I could do with micropipettes was really, I could really overpass a lot of challenges of how to place a cell, how to take it, and how to locally press it, for instance. And then problems with this is, I do a lot of experiments nowadays. I still do a lot of experiments, so I could have so many issues. I mean, it's difficult life to do experiment with cells, from the culture to the handling to the knowing how they feel, why there's so much variability. But I could say that, how can I say? Nowadays, I'm really, I think it's pretty exciting, and I don't feel it as so much as a challenge, except that to let's say I really love it in a way that now, once you get to know all these challenges, you overpass until you get into a situation where it's not about just suffering with this, but finding new tricks to overpass new challenges. So I don't know, I probably got lost, but in any way, I think there is a lot of technical challenges to manipulate microscopic objects. But there is so many tricks that even yourself or people did develop that a lot can be done, even though, of course, there is so much yet to do. I was just mentioning this temperature control is hell, but you can either just work at room temperature and do a lot, and you can still develop a lot of new tricks to locally control temperature as this is done in atomic force microscope, for instance. I don't know if this answered the question, but you tell me.
Muchas gracias.
De nada, gracias.
Y bueno, gracias por compartirnos tu trabajo.
You're welcome. It was a pleasure. Gracias, gracias.
Bueno, vamos a continuar con el siguiente ponente que viene del Centro de Medicina Regenerativa de Utrecht, en Holanda. Es el doctor Ricardo Levato. ¿Ya nos está viendo? Súper. El doctor Ricardo se dedica principalmente a la bioimpresión aplicado en medicina regenerativa. Entonces tiene una ciencia aplicada para todos nosotros. Y doctor, usted puede comenzar.
Perfecto, muchas gracias. Buenos días a todos. Haré la charla en inglés, pero bueno, si hay alguna pregunta en castellano también no hay problema. Dame un segundo que voy a compartir la pantalla. Vale. ¿La veis en el modo correcto o es en...?
Sí.
Sí, perfecto. Ok. Well, first of all, thanks a lot for your invitation. It's a pleasure to be able to present the work we have been doing here in Utrecht to this audience. As I mentioned, as the introduction said, we work more on applied technology rather than biophysics, but we have some tools that may be of interest also for you as well, of course. Briefly, something about our lab. So we are based in Utrecht in the Netherlands. We're actually based in the Utrecht Institute building. So we share a lot of lab space with biologists that focus a lot on organoid biology. We primarily develop tools to pattern cells and ECM materials in 3D using different bioprinting technologies. I will focus specifically on one of them. But we do all sorts of extrusion, electrowriting, and light projection printing as well. And we also develop some materials, as you will see, that we use as models to mimic different aspects of the cellular matrix and how cells interact with it. In terms of application, we focus primarily on musculoskeletal system and on soft tissues such as the liver and the pancreas, and I will give you some very specific examples today. First of all, this is why we do this work. Of course, the ultimate ambition is to regenerate full organs in the lab and then use them for transplantation. But that's a bit of a still far away goal, so we're more focusing also on developing in vitro models of different biological phenomena. And I'd like to start from this video of the development of the vascular system of the zebrafish embryo, just to remind us that cells have all the instructions to create different parts of an organism and the full organism inside of them. When we culture them in the lab, we can only recreate very small portions of these tissues. And one key challenge is that when we culture cells in 3D, typically in these hydrogel systems, what we start with is actually, this is an extruded filament of a fairly high density of cells. So we have 100 million cells per ml, which is quite comparable to most soft tissues. The cells are still quite far apart. And that after printing, there is a whole need for cell remodeling and tissue remodeling events to take place. So printing is sort of a way to give us tools to accelerate how this maturation events post-printing occur, and how we can control certain key signals within the matrix. Specifically, I will talk about how we can use pre-assembled cellular structure, for example, organoids, as building blocks to create larger tissues. And of course, that's the area of an advantage in the sense that the cells already acquire some key elements of the organ you want to recreate. Of course, our tissues don't sit in the void. The cells interact with different cell types and with different tissues in the body. So if you think of a muscle, for example, is connected to a tendon, and also vascularized, of course. So we need technologies that allow to pattern multiple set types at once, and that's what we will also talk about. And finally, the matrix where cells live, cellular matrix, the ECM, is not just a structural support, but is also a depot of different tethered factors, morphogens, and growth factors. So we also study techniques to bind and load growth factors in specific areas of our 3D tissues. So starting from the first part, let me first introduce the technologies we use. You may be familiar with extrusion-based printing, which is what you see here on the left hand, where essentially you have a hydrogel filled with cells that is smeared through a nozzle onto a surface, and by drawing layer by layer, you create a three-dimensional object. You can also use light-based technologies, most commonly stereolithography or DLP, where essentially you have a vat of a light-responsive polymer, which can be also a protein or a carbohydrate if you want to make the ECM. We send light specifically on certain spots, and only where the light hits the polymer becomes a solid. And then by fishing a first layer out of the solution, we can introduce a second layer of material and keep printing until we obtain our object. The technology I will focus the most, though, is something my lab introduced in the field of biofabrication already 5 years ago, called volumetric bioprinting. This essentially entails shining light onto a wall volume and having your object being created in a matter of a few seconds. The way it works is like this, as you see in this real-time video here on the right hand. Sometimes when we are online, it lags a bit, so I hope you can see it at the right speed. But essentially around the 12 seconds mark, so now you should start seeing appearing a tower of the Church of Utrecht. So that's the landmark of our city. And essentially this is a technology that allows to create objects all at once instead of the classical layer after layer techniques. The way it works is that we send visible light, in this case blue light, onto a digital micromirror device, which encodes 2D projections, and sends this 2D projection onto a vial containing a photopolymer. In our case, most often we use the collagen derivatives, gelatin methacryl oil, gelatin norbornene, as you will see, but any sort of protein modified to be photoresponsive. And the vial starts rotating, and with every angle of rotation, we send a different image, so that the composition of all these different images together makes a tomographic reconstruction of the object we want to print, much like what happens when you make a CT scan to see your bones at the hospital. But instead of sending X-rays, we are sending visible light, and instead of imaging, we are now printing. In real life, it looks a bit like this. To start the video myself, sorry. So what we load the vial containing, this is gelatin methacryl oil. We load it in our printer. Normally it will be sealed, of course, if it's a sterile print. Rotation starts, the light turns on, and then we quickly see how the object is being printed. The unreacted material can be washed out. The cells that are there can be replated or used for different control experiments. And essentially the object that we want to print, it's retrieved. What is happening here is that we're playing a bit with the photochemistry of the material. So the gelation chemistry has this type of curve typically, and only above a certain irradiation dose, we do have actual cross-linking. So we make sure that all the parts we want to appear within our volume receive more light essentially than the background, so that we only have cross-linking on the areas that we really want, and everything else remains unreacted. And therefore it can be eventually washed out, as you will see in a second in the video. And essentially you retrieve your part and you're ready to do your experiments. So this technology is, compared to other printing techniques, is extremely fast. So if you use conventional layer by layer printing by extrusion or digital light processing, if you want to make this oracle model of 4 centimeter cubes, it can take anywhere between half an hour to a couple of hours. But with volumetric printing, it's always within 20 seconds, more or less. In addition, when you do extrusion, you will have these filamented patterns on the surface, or DLP, or these pixelated patterns. In our case, we get fairly smooth surfaces thanks to the rotation of the object. And because we are printing with light, we can print across existing objects. So you can make a movable part, as the example, this fluidic valve, where you have a ball that blocks the flow on one side and permits the flow on the other. So the ball is printed afterwards inside this already existing cage. In terms of resolution, we can print down to 40 micrometers roughly. and perfusable channels as with about 80 micrometer wide. The very first cell print that we made was this bone model. There is a patient-derived STL scan, STL file, and everywhere that you see here in this CT reconstruction in white, we actually it's a gel which is laden with osteoblasts derived from MSCs, which are here stained in magenta. And you can also make secondary seeding or printing to introduce, for example, other cell types, like in this case, endothelial cells for making blood vessels and support cells to make the pericytes that stabilize these vessels. We also show that if you print, the cells that you print can be kept alive for very long. So we culture for more than a month, in this case up to 1 month with these articular chondroprogenitor cells, which are highly viable. And over time, they produce their own cartilage-like matrix, making this meniscus construct to acquire similar mechanical properties in compression as the ones that the human menisci will actually have. So about 300 kilopascal in terms of mechanical stiffness under compression. Everything I showed now, though, is you have cells scattered around, which is fine for cartilage, but you have essentially primarily an ECM-rich tissue with few cells encapsulated and isolated here and there. But in most other soft tissues, for example, you want cells to connect and reorganize with each other. And what you see here in this monolayer culture of endothelial cells from the Trepat lab, epithelial cells from the Trepat lab, that are now forming these pits and crypts mimicking the layers of the intestine. So we come up with the idea of using these organoids forming cells also for printing. And this is something that has also been explored in the recent years by the Lutolf lab, formerly at the PFL, now they are at Roche, where they were extrusion printing this stem cells from the intestine into filaments that eventually create this luminized intestine-like filaments that do have cryptin villi. However, extrusion printing is extremely limited in what you can produce. And most of this structure are fairly large. So if you want to print with good resolution, you will probably end up damaging these organoids while you're printing them. In our case, we are more interested into liver organoids because some key applications are in the areas of drug screening. And acute drug-induced liver failure is the main cause for drugs being withdrawn from the market, even after approval through clinical trials. And unfortunately, the reason that many drugs actually fail in clinical trials or even after, it's because there is only poor predictive capacity of current animal models, but also in vitro tests. And we can culture hepatocyte in vitro, but they differentiate and die very quickly. So we can only do acute assays and no chronic exposure seriously, essentially. So we use this, as I mentioned before, we collaborate a lot with the Hubert Institute, where also Hans Clever was before moving to Roche as well. So there was now a cluster of expertise at Roche. But their lab in 2015 isolated bipotent progenitor cells from the liver, from liver biopsy. And these cells, when cultured in 3D and matrigel, typically are able to form these cystic structures, which with the right media can be differentiated into either hepatocytes or cholangiocytes. And of course, in our case, we're interested in hepatocytes. We originally tried the approach of just extrusion printing structures that we can then place in some perfusion bioreactor to play with the porosity a bit. But what we quickly realized is that these organoids can be even 300 micrometers in size. When you want to print them through a nozzle, there is a lot of mechanical stress involved. So you do get quite a fairly amount of cell death and destruction of these organoids as well. So we thought our technology, our light-based printing technology, could avoid this problem, provided we solve another challenge first. So that when you do light-based printing, essentially you are aiming to deliver light very precisely within your photopolymer, but cell scatter light. And that can, of course, mean that if you try to print this simple yin-yang model, for example, with single cells or organoids at the density of already 10 million cells per ml, which is not especially high, apologies, we get these shapes which kind of resemble what we want to make, but they're not very precise. So we came up with this solution where we can use a biocompatible contrast agent called iodixamol, which essentially matches the refractive index of some key organelles within the cells. We typically aim to match the nucleus, which is the main scatterer. So with this, we essentially make cells partly transparent to the light we are sending, and we are now able to restore fairly well the geometry we intended to print. So having solved this challenge, we move forward. That's the work of Paulino Núñez Bernal, formerly a PhD, now a postdoc in my lab. By the way, an extremely talented Mexican PhD postdoctoral and now doctor. She has been working on this approach by taking these preformed organoids and re-embed them in our gelatin gels and perform printing with different techniques. And indeed, she confirmed that by using a volumetric printing, because you have only light and no mechanical stresses, we can get we can better preserve the shape and the viability of these organoids, which eventually grow to larger size over culture simply because they are starting from a better point from the beginning. Whereas if you do just extrusion or also just micropipetting to cast the hydrogels, you have quite considerable cell death and smaller organoids in the end. It's not just a matter of size, of course, because the fact that we're not disturbing the culture process of these organoids also means that they have more time to mature. And indeed, we observed that in the volumetrically printed group, we have indeed a formation of a polarization within our organoids. So in the lumen, we have expression of MDR1, a ion pump normally used to transport drugs from the basal side of the hepatocytes to the apical side, which apical means is the part that faces the bile ducts to excrete product in the bile ducts. And this is something that we observe here in our organoids, so that we have a lumen in the beginning, which in the inner side, which is the bile duct side with these hepatocytes, and the rest on the back. And we have albumin production, so we have the differentiation into hepatocytes, and some liver enzymes are also better expressed compared to controls in matrigel. Overall, so therefore we see that we get better polarization, better maturation of the organoids, which is quite encouraging. And so we can start now playing with the printer to see how external geometry of the environment in which we pattern our organoids plays with the function. So we designed these different patterns of gels, which is called gyroids, that are geometrically defined, so that we have some fairly complex paths when you inject flow through them. We have fairly convoluted flow profiles, and we have more straight and simple flow profiles in the other type of geometries. So we use this to test how these different geometries indeed influence organoid functionality. And we focused on the urea cycle. So we normally, the liver takes ammonia, a toxic byproduct of protein metabolism, and then converts it to urea that can then be excreted by the kidney. So we produce these objects. The printing, as I mentioned before, is very fast. So within 15 seconds, we obtain every object. So in 2 minutes, we can fill a 12-well plate and then connect this to the perfusion system. And indeed, we observe that when we have the more complex geometry and porosity, the residency time of the solutes is higher. The shear stress is also a bit higher and stimulates better the organoids, and we get therefore faster elimination of ammonia. So just to remind that we can now create these chips where we can optimize the functionality of our cells for drug screening purposes, but also eventually for transplantation purposes, for example. Of course, no tissue is completed without vasculature, so we are focusing a lot on how we can actually introduce vessels in our systems. And Maria Mgei is working on these co-cultures of endothelial cells and mesenchymal stromal cells. And when you culture these spheroids together in the right gel, they form this very pretty network of capillaries, which eventually, when you apply flow, they can also open up, and you can have indeed perfusion through them. However, there is a caveat here, and this nice picture is done in matrigel. And everybody that works with biology loves and hates matrigel. Loves because most time it works when you have the right batch. Hates because batch to batch you have about a 60% variability. And therefore, ideally, you would want to do this in a chemically defined matrix, potentially, which you can also print, so they can introduce the advantages of controlling architecture as well. So This was the task that another student in the lab took on. His name is Mark Falland. And essentially, it took our classical gelatin, so a collagen derivative, functionalized with methacrylate groups, so we can still photocross-link, but then also add adamantane and mix with activated cyclodextrin, so that we can create also supramolecular bonds on top of our gel. So what happens is that the adamantane is this hydrophobic molecule that likes to hide inside this bucket, the cyclodextrin. And this bond is completely reversible, so it keeps forming and reforming all the time. The nice thing is that this makes our gel a bit stiffer, so that macroscopically we can print and preserve better the shape of the object. So we can see here this disc are able to self-stand, whereas if we don't have the hybrid bonds added to the acrylates, we just have these very soft gels here. But from the macroscopic level, from the cell point of view, because these bonds can be broken and reformed very easily with low forces, cells are able to migrate and grow. For example, these are endothelial cells GFP stained that form these networks across very large objects. So here we have 500 micron, as you can see. So this is several millimeters in size. We can get nice vessels forming. We screen a bit how the optimal ratio between the covalent and the hybrid and the supramolecular bonds allows better endothelial cell vessel formation, and eventually settle to this specific formulation where we have 60% covalent bonds. So 60% of the amine of the gelatin are converted to acrylates and 40% to the adamantanes. Because these gels allow cell migration, we can then also use them to perform immunoassays in 3D with engineered T cells, for example, CAR-T. And in this case, we encapsulate breast cancer organoids in different gels, so Matrigel, our covalent gel, and our hybrid gel, and you can already see how the T cells like to migrate inside the hybrid ones. So in the Matrigel, we have poor migration. In the covalent, almost nil. And yeah, when they started the experiments, there is massive inflow of T cells within our gel. They're able to migrate, and they start attacking the tumor. As you can see here, the yellow slowly disappearing and disassembling. So with this, we can now create models where we print healthy spheroids, as you can see here in yellow in this cartoon. Then we insert in a specific spot the breast cancer, seal it off, turn the object around, then see the T cells and see where they locate. So if they're able in 3D to navigate these gels and find the tumors without attacking the healthy cells. And this is exactly the case that we see. So most of the T cells are actually infiltrated into the tumor core, staying here in red. They start attacking it. As you can see, also the cell death in the tumor due to as marked by activation of caspase. So we have some apoptotic markers showing that the cells are indeed dying. So this can help us to create a 3D complex labyrinth to test different CAR-T cells in chemically defined gels, which is quite promising by itself. So I promised at the beginning that I was also going to touch base on how we can actually introduce multiple materials in our prints. This is actually fairly easy when you do extrusion technologies, because you can just have on your printer a syringe with different cell types or different materials and alternate how they're printing. When light-based printing is a bit more complex. So we thought we could actually use two approaches. So we can either combine printing with pre-existing objects, and that's with electrowriting, I will tell you in a bit. But first and foremost, we can actually combine the advantages of light-based printing with extrusion-based printing. And to do that, we use, instead of using the classical gelatin materials, we use printable materials made of microgels, which are these granular microstructures, so that they can fuse together, and then you have some microporosity in between. But before cross-linking, this behave a bit like sand, so you can actually insert a needle in it and perform extrusion in specific areas. And this was the work of another student in the lab, Davide Ribezzi. So we can also use light to anneal and glue these particles together, as you can see here. So we can do our prints again, but this time they are composed of tiny gels attached to each other. And if you can appreciate the porosity, I hope the resolution comes well across the screen. We also saw some little tricks, because of course these gels also scatter light a bit. depending on their temperature, they can be more transparent, so that makes us able to print a bit better. You can control the mechanical properties and create different type of structures with channels, tubes, and so on and so on. The advantage that these microgels are also inherently porous, so the interparticle porosity is a void in which cells can infiltrate, and also media, as you can see from this colored dye. And for example, if we take here neurons, we put them in a classic bulky gelma. They are quite rounded, they don't like it especially. But in the microgel, they're able to find a space in between the gels, migrate, and form these nice axonal connections. The same works for vascular networks produced by endothelial cells. And because our gels are also a bit softer than normal ones, we can also trigger adipogenic differentiation of MSCs. And that's a bit for a different application. As I mentioned, we can print multiple cell types, but the most appealing part is that we can now combine extrusion with volumetric printing. So in this example, we are printing some very simple rings of cells on top of each other, then move it to the volumetric printer. So we can superimpose a light pattern in 3D. I will eventually form whatever object we want, having embedded these rings. And you see here some examples of this Saturn shape object, this organoid in the middle of a ramified vessel, and the head of Einstein with a small portion of his brain in there. The advantage here is that we can really go locally at very high cell densities without having much scattering problems. So we have about 50 million cells per ml without having to use the refractive index matching approach. And we can now start tinkering to make more complex models. Another approach to multi-material printing that I mentioned is, of course, use this extrusion approach. To give an example of what we use it for, we actually extruded some pancreatic beta cells, which we specifically engineered to express nano-Luc as a reporter of insulin, and also to express melanopsin. So we actually use them for optogenetic control of insulin release, so that we can illuminate with blue light and trigger the cells to release insulin on demand, essentially. We first saw that these cells actually function only when they are clustered together at higher densities. So the extrusion printing is very handy here, because if you just suspend them in the gel, they work very poorly. And then we did some co-culture of these beta cells printed in a ring together with mesenchymal stromal cells. And we observed how we can use actually the insulin release to trigger the adipocytic differentiation of these cells. And indeed, we can create these co-culture models where cells are located in different areas of the print. I mentioned before, we want to do another approach to do multi-material prints is also to first create pre-existing objects and then use light to print around them or through them even. We can do that also with opaque microfibers that we produced by melt electrowriting, which is essentially a sister technology of electrospinning, marrying electrospinning with extrusion printing, that you can make these very small micrometer-sized fibers and pile them very precisely next to each other. So we make some tubular stent light-like fiber objects, put them in the vials of our printer, illuminate, and obtain different structures. We need to do some software corrections to correct for the attenuation of light caused by different geometries, but that's all quite mathematically derived, so we can easily calculate how to change our projections. And we can now use these fibers to reinforce mechanically our prints. So if we try to print, for example, a tube with these gels that cells like in order to grow, they're fairly soft, so they will collapse under their own weight. And when we add increasing amount of these fibers, we get macroscopically stiffer gels, even though the gel itself, so the cellular microenvironment is still fairly stiff. But macroscopically, we can now match the mechanical profile of pig coronaries. So we have a quite promising mechanically functional structure. We can introduce occlusions, valves, or anything we can print, because of course printing gives us the freedom to control the geometry. You see here in red the printed gel with the venous valve, and in green you can see the fibers on the outside that are making the scaffolding of this object. And we can also repeat the print multiple times to pattern multiple cell types. So you have, for example, endothelial cells in the lumen, and then two different stained HMSEs as a model of different layers of the vessel. The final application I wanted to show you is also the generation of gradients or patterns of morphogens or biologically active molecules. And for that, we use a different type of photochemistry. We use a thiolene click chemistry, where we have a reactive norbornene on our gelatin, which essentially react with any thiol. So any protein that has a free cysteine can now be cross-linked to our gel. So what we can do now is we use the thiol cross-linker, for example, DTT, to print our objects. Then we infuse with the protein of interest, and we make a secondary print that now will bind the protein only where we want inside the object, and then wash, and we have our print. If you do things right, it looks a bit like that, and that's the work of Mark and a student in the lab. What you have in green here is a simple tube, a cylinder with a tube in the middle. What you have in red is not a physical object, is a pattern of a grafted fluorophore. We can actually do quite fancy structures, so we can reach a resolution of about 50-60 micrometers with the patterning. We can pattern all sorts of geometry you want and combine with complex print and patterns specifically in a very nice way. So it gives a lot of freedom to control both geometry and chemistry of the matrix that we use for culturing cells. Now, why does this matter? Because, for example, we can now study or guide cell behavior in 3D at very large distances. So this, of course, was already possible if you use, for example, photomasks or two-photon polymerization, but you would never be able to do that across centimeter thick objects, whereas this technology that we have developed allows to do that. So, for example, we have here a tube, again, with the UVEX inside, so we make the internal lining of the vessel. And then we pattern VGF only on one part of the object, and we can see how endothelial cells can penetrate through our gel and start migrating only in the region where we have the grafted growth factors, and in the other they just stay on the tube and don't do anything essentially. We can also quantify how much the cells migrate over time, and the nice thing is that this migration happens both if you deplete VGF from the media, so the only VGF is present in the matrix, but also if you have excess of VGF in the media, this selective migration occurs only in response to the tethered growth factor to the ECM, which is also quite interesting to study how we can use this to guide cell migration specifically. In synthesis, I want to introduce a bit this new technology called volumetric printing, which is a layerless, nozzle-free, light-based printing technology that allows to produce very complex geometries very fast, very quickly, in a matter of a few seconds. We can combine multiple cell types, microgels to introduce porosity and combine with extrusion printing, but also we can print across existing objects and therefore combine with other prefabricated structures for mechanical reinforcing or any other application. And finally, we can create 3D patterns of morphogens, which is quite interesting for studying many developmental approaches, but also regenerative medicine approaches as well. Finally, I also wanted to remark that I talk a lot about this part of the equation, volumetric printing. Is not the only technology out there. And in our lab, we don't believe that there is one technology which is a magic silver bullet that will solve everything. Most likely, you need to combine techniques together in order to overcome the limitations of a single technique. And especially if we want to create fully functional tissues, we need different techniques that allow us to improve mechanics, to integrate multiple tissue types, to provide the vessels and nutrient supply, to mimic tissues, but also to upscale the fabrication. And that's where volumetric printing is especially useful. And this is true both for regenerative medicine application, but also for making in vitro models, of course. Finally, let me thank my lab, so the people that actually do the work. I just have the pleasure to talk about it. And I'll be happy to take any questions you may have. I can't hear you.
¿Tenemos alguna pregunta? Perfecto.
Thank you. Very impressive work. So I guess I have a couple of doubts from the principles of the technique.
Yeah.
So one of them, I was not sure why you needed to rotate. I didn't understood it. And the second one, I'll just say the two at the same time. And the second one is also I didn't quite understand how come you don't get polymerization like in the area between the wall and the center of the feature that you want. I don't know if it's like because it's not a 2D photon thing, right? So is it just like focusing of the light, or is it like a holographic projection?
Yeah, is physically is not an hologram, so it's not really an holographic projection. But I'll start from the first question. So the rotation, because you need, we are making essentially a tomographic reconstruction. So we have two options. Either we rotate the light source around our object, or we rotate our vial. You can avoid rotation if you have many light emitting paths that send things in parallel. That would also work. But because we only have one light source, the only way to illuminate from every angle, that's what you need to do tomographic reconstruction, is to rotate either the light source or the vial. And in our case, it's easier to rotate the vial. As for the second question, what we do is not as I said, not an hologram. It's literally a tomographic reconstruction. That means we send filter back projections of the object we want to print at every angle. And they overlap in the areas where we want the object to appear. So everywhere is illuminated, is a single photon process, it's not a two photon process. Everything receives light, but only, like in this video, only where you see the blue fluorescent pattern appearing. That's where the light dose is high enough, in this case, to show fluorescence, but is also high enough to trigger cross-linking. And the way this works specifically is that the cross-linking reaction is not immediate. It's not the light is on and cross-linking happens. It builds up over time, and only when you reach a certain light dose, the polymer solidifies. Otherwise, it stays liquid. So essentially, everywhere is illuminated, but here light comes faster. So by the time we stop the print, this part has received enough light to cross-link, and the rest not. Which also means that indeed, if you were to keep the printer on for too long, everything will be polymerized at the end.
Awesome. Thank you so much.
Yeah, you're welcome.
In YouTube, have another question. And is, have you been able to construct perfusible capillarities within the printed structures?
Yes, so the resolution of the printer is about 40 micrometers. So that's the smallest we can make. And as I was mentioning at the beginning, this is only when you make positive features, so actually pillars, spikes, or other elements like this. Open channels, the actual resolution is about 80 micrometers. So that's the smallest we can make, while still having an open lumen and pattern channel. Then you need to endothelialize, of course. So that this 80 micron is already a bit too big for being capillaries. Capillaries, we still rely on cell self-assembly. So the idea is that the printer gives us the chance to create the larger channels, and then you have the smaller 10 micron, 5 micron capillaries made by cells by themselves, which is also what we try to do over here, essentially.
Okay, thank you. And another question, I'm gonna do in Spanish.
Vale.
Es respecto a las consideraciones de diseño que tú tienes para el cultivo de los organoides.
Sí.
Justo la mención que tú hiciste para el desecho de nutrientes y para la disponibilidad.
Vale, o sea, si entiendo correctamente, Me estáis preguntando qué criterios de diseño tenemos para asegurarnos que los organoides reciban nutrientes y puedan funcionar, ¿correcto?
Sí.
Vale, en primer lugar, hay que considerar que, bueno, aquí no hay vasos en este caso, no hay transporte activo de nutrientes a través del gel. El gel es una barrera de difusión, o sea que retrasa la difusión de moléculas según su peso molecular a través de la estructura. Típicamente en la literatura se lee siempre, la célula no debería estar más de 200 micras más lejos de un vaso, de un capilar. En realidad, con estos geles que son bastante permisivos, 500 micras aún sobreviven bastante bien. Con lo cual, en este caso, imprimimos, de hecho, estas estructuras que veis aquí um, son aproximadamente unas 500, 700 micras, de manera que cuando un organoide está dentro, las células en el exterior, también en el medio del organoide, no estén a más de 400 micras del borde, que es donde inyectamos el medio de cultivo. Entonces, típicamente minimizamos la anchura de cada elemento que imprimimos para que las células que estén adentro no estén demasiado lejos del frente de difusión. ¿Está claro?
Muchas gracias.
¿Nada?
Y una última.
¿Sí?
En las bioimpresiones, en donde tú embebes las células,
Mm,
bueno, así lo entendí,
sí.
justo este tema de difusión, ¿cómo haces
Mm.
para que las células sobrevivan o cómo tienen que ser esos materiales para que permitan el proceso de difusión y se mantenga la estructura que tú bioimprimiste?
Vale, o sea, si me estás preguntando durante el proceso de impresión, con esta tecnología no es un problema porque realmente el proceso tarda unos 10 segundos y entre el tiempo que, o sea, mezclamos, básicamente haces un cultivo celular normal, recoge, sea en 2D, si son células 2D, si son organoides, en matrigel o otro material parecido, recuperamos los organoides o las células y las resuspendemos en nuestro gel. Se carga dentro de la vial de la impresora y imprimimos y todo el proceso desde que resuspendimos y imprimimos será muchos 5 minutos. Entonces en ese tiempo las células aguantan bien. Em, Después del cultivo hay que considerar es lo que te decía antes del tamaño de la porosidad y las estructuras que imprimimos, porque desde luego si el gel es demasiado ancho, es verdad que pues los organismos recibirán menos nutrientes y padecerán a lo largo del cultivo. Pero en el momento, en el tiempo de impresión, esto realmente no es un problema, porque la impresión es tan rápida que no vemos ah, ningún problema de viabilidad o falta de nutrientes en ese tiempo.
Muchas gracias. ¿Y tú, hay alguna otra pregunta? ¿No? La última, en tu trabajo, ¿tú has llevado a implantes tu trabajo? O sea, ¿los has imprimido
Ujú.
y utilizado en algún organismo como tal?
Sí, no lo he presentado hoy. Hay de hecho, esta imagen que tenía aquí al final, es de un trabajo previo. Esta es una estructura polimérica, pero no tiene células aquí, que usamos como soporte de hueso. Y aquí una parte de cartílago que sí que tiene células con rocitos. Eso, eso es de tamaño de conejo, lo pusimos en la articulación humeral del húmero de la espalda del hombro, perdón, del conejo. Y observamos efectivamente una regeneración de la parte de hueso, pero no la de la parte de cartílago, con lo cual al final la funcionalidad de la estructura no funciona tan bien. Em, Y ahora mismo no llegué a presentarlo porque es un trabajo un poco temprano, pero estamos trabajando en estructura parecida a la que enseñé de hígado, o sea, geometría parecida a estas, pero encapsulando organoides de páncreas, concretamente de islas de Langerhans. Y esto, estamos empezando ahora un implante en ratones diabéticos, pero aún es un poco temprano. Espero poderlo presentar con resultados el año que viene o la próxima vez que tengáis este tipo de eventos.
En el siguiente simposio.
Sí,
Muchas gracias por
de nada,
compartirnos tu trabajo.
de nada. Buena continuación.
Eh, ya se fue. Bueno, vamos a continuar con la última ponencia de este primer bloque. Aquí ya no tenemos físicos, tenemos una ingeniera en biotecnología. Ella es Ana Zatarain. Ella es ingeniera en biotecnología, estudió en el Instituto Tecnológico de Monterrey y actualmente se encuentra realizando una estancia doctoral en el Instituto de Perinatología. Su investigación se centra principalmente en el uso de células tronjales para terapias regenerativas enfocadas en lo que serían enfermedades del sistema nervioso, particularmente los defectos en el cierre de tubo neural. Y bueno, los dejo con nuestra ponente.
Gracias. Hola, sí, listo. Muchísimas gracias. Bueno, me habían comentado que presentara en inglés. The organization of tissue across embryology. And I'd just like to point out 3 important things to consider when talking about these morphogen gradients. So first is competence, and what I mean by this is that we'll have a cell that is secreting these morphogen gradients, and they'll be diffusing across specific tissue. And we need, for this to work, we need our cells that are receiving the information to have the capacity to do so, and then also to allow for a downstream cascade of information so we can express the genes related to this signaling molecule. And then I'll also point out how important context is in development. So we have relatively few signaling pathways that regulate the whole of embryology. So it needs to be very specific, and the information that is being received will be interpreted differently depending on the time and the place of a specific cell. And then finally, just to mention how complex these regulatory networks are, and they have this very fine-tuned, well, they're controlled in a very special manner so we can produce the actual embryo. And I'd like to continue talking about these gradients, but focusing on neurulation. So neurulation is a process where the neural tube is formed. This is the precursor of the central nervous system, and it is governed by these morphogen gradients, which are generated throughout the whole of this tissue. So we can find 2 basic axes alongside it. So we'll have the cranial caudal axis, as you can see on the first image, where we can find a high concentration of retinoic acid at the cranial part of the tube, and then a high concentration of fibroblast growth factors on the bottom. And these 2 signaling molecules will generate a gradient across the tube, allowing for cells to begin specifying into specific neural progenitors. And then if we cut across the tube, we can find the second image where we'll see a second axis, the dorsoventral axis, where we'll find a high concentration of Wnt at the top and Sonic the Hedgehog signaling molecules at the bottom. And this gradient will also help our tissue to become more specialized and our cells to also become more specialized in their neural sulfates. And this will allow for the tube to subsequently form the brain and then the spinal cord. And a way that we can better observe and understand this is through the French flag model, where it basically states that the morphogen concentration begins at a high rate, and it diminishes across time and space in an exponential manner, depending on the space it is trying to move across and the decay rate of our specific protein or molecule. And so what this French flag model tells us is that we'll have specific clusters of genes that are expressed depending on the morphogen concentration. So we'll start with a high concentration, and we'll see a cluster of genes being expressed related to the blue color of the French flag. And once a specific threshold is no longer met because the concentration is diminishing, we'll have the expression of a second set of gene clusters visualized in white, and then same thing with the red color and the red cluster of genes when the concentration is minimal. And I'd just like to talk a little bit about neurulation. So this process starts around the 3rd week of gestation in humans, right after gastrulation, and we can divide it into 2 types or 2 phases actually. So we'll have primary neurulation, where it starts with the inhibition of the BMP, the bone morphogenesis protein signaling pathway, where the center of the ectoderm will begin to differentiate into neural cells, which will form the neural plate. Due to molecules, these morphogens that are expressed by the notochord, right there in orange. And so these cells will begin proliferating at a very high rate, leading for them to fold and form the neural group, which will subsequently close in the last stages of neurulation, forming the hollow tube, which is called the neural tube. And then we can also consider secondary neurulation, which occurs on the caudal part of the embryo, so on the tail bud, which begins from a specific type of independent cell populations called the neuromesodermal progenitors. These cells have the capacity to differentiate into either neural or mesoderm, depending on the morphogen gradients that they are exposed to. So at high retinoic acid concentrations, we'll have these progenitors differentiate into neural cells, which will begin migrating into the center, forming the hollow tube, which will subsequently fuse with the primary neural tube. And then we'll also get some mesoderm when the neuromesodermal progenitors are exposed to Wnt signaling molecules. And so as we can sort of understand, these morphogens create a very delicate balance between all these concentrations that are expressed and that are leading cell fate. And so we can find that if we have some sort of disruption of this balance, this can result in neural tube defects, which are really catastrophic. And depending on which part of the neural tube we'll find these defects is the type of, again, defect that we'll see. So on the cranial region, we'll find defects of the brain, where the brain doesn't develop, or some parts of the brain don't develop, such as anencephaly. And then we'll also have defects where part of the brain develops outside the body in this herniation of tissue and skin, such as anencephalocele, as you can see right there. And then also, if our defect can be found on the spinal or caudal region of the neural tube, we'll find spina bifida, out of which the most common are meningocele and myelomeningocele, where the neural tube and the spinal cord develops outside this body, also in this sort of herniation that you can see in green. And so these kinds of defects are really important because they're the most common types of birth defects. And they're also associated with a loss of motor functions, several discapacities, and it can be very hard for the families when taking care of them because most of them have a rather low life expectancy. And so if we can understand these molecular mechanisms driving neurulation better, then we can also understand how it is that these defects occur and perhaps try to fix the damage done. And shifting gears a little bit, I'd just like to take a little bit of time to talk about stem cells, because these types of cells really help us to model development, regenerative biology, and also model diseases. So stem cells have the unique capacity to self-renew and differentiate into specialized cells. And so we can harness this to build three-dimensional tissues that help us mimic in vivo realities and try to study them. And a really interesting innovation was done by Yamanaka, I'm sure you all know, where he found that using 4 transcriptional factors, he could take adult cells or somatic cells and reprogram them into induced pluripotent stem cells, which have the capacity to regenerate the 3 germ layers, as you can see. And then also this helps us model diseases, study development, model organoids, but then also helps us try to evade the use of embryonic stem cells, which, as we know, have certain legal and ethical connotations. And so the final part of the introduction, I'd like to talk about organoids. So if we can mimic the gradients that we talked about earlier, on the morphogens, we can obtain self-regulated 3D structures that help us, study development. So we can do this from taking different types of stem cells depending on what it is that we want to study. Thank you, gracias. And then we can also use synthetic matrices to study cell and extracellular membrane interactions to allow for the diffusion of nutrients, growth factors, among other things that cells need to survive. And then we can also use these to build or actually use soluble factors to help drive these cells to differentiate into the tissue that we want to study. And so a really interesting technology that's been happening these last couple of years is the meshing of engineering and biology to build organoids on a chip, where we can develop microstructures that help us generate these morphogen gradients and allow for these stem cells to differentiate into organogenesis and even to study organs such as liver, heart, et cetera, et cetera. And I would just like to finally point out research that's been done on neural tube organoids. So they're very interesting, and researchers have been able to model the morphology of these of the neural tube with the bending, folding, and closure of their organoid. And as you can see, they've been able to characterize their cells with red being neural cells with N-cadherin, and then the ectoderm in blue with E-cadherin. And then also, they've been able to determine specific cell populations found inside the neural tube. So neural cells with PAX6 in green, and then in pink, the neural crest cells. And although there's still a lot of research that needs to be done so we can model diseases and actually get the morphology right, there's a lot of innovation that we can do, and we can use biomimicry to build better organoids. So if we understand the way the neurula is structured and the neighboring tissues alongside it, we can build better organoids that simulate the actuality of in vivo neurula. And so, for example, we can see that the notochord, which is sits right below the neural tube, secretes Sonic the Hedgehog signaling molecules. So if we understand this, we can also build organoids and micro devices that simulate this. also how the ectoderm secretes Wnt signaling molecules above the neural tube, and then finally how the mesoderm and the somites specifically secrete retinoic acid and fibroblast growth factors alongside the whole of the neural tube here in white. All right, so understanding all these aspects, we've tried to develop a stem cell culture medium to mimic neural tube development and closure. And we've tried to separate it into the primary and secondary neural tubes, so we can also model diseases associated to each of these parts. So we start with neural induction and neuromesodermal progenitor induction, followed by the actual neural tube model, trying to build these morphogen gradients that we talked about. And some of the issues that we came across when we first started to build this organoid was first that we wanted it to actually be three-dimensional, because obviously a two-dimensional structure couldn't really help us to understand the morphology and all the changes that occur. So we definitely decided upon an organoid. And another issue that we came across is that we wanted to have all four of these signaling molecules come into the organoid in a concentration gradient, so have this exponential gradient come across it and feed the organoid. And when we tried to simulate this, it was just a mess. So we were having like all these molecules. We had the medium supplemented with with the signaling molecules coming into the culture chamber, and it was just a mess. It was mixing everywhere. So we finally decided upon the use of hydrogels. So as you can see, the idea is to have our neural tube organoid from induced pluripotent stem cells and have it encapsulated by this hydrogels membrane. So this would allow for the diffusion of our signaling molecules and have this gradient that we talked about, while also providing structure for the organoid and then being flexible enough for it to grow, because as we know, during neurulation, the neural tube elongates. So we also need to accommodate that. And so I just want to show you guys real quick the first prototype of our microfluidic device model. So we opted for a 3-layer device where we'll have the neural tube organoid growing in the middle chamber. So we'll have it here in its hydrogel capsule, and then we'll be feeding it with the FGF8 and retinoic acid signaling molecules across these microchannels, and then they'll be flowing across this triangle shape, which is covered also in hydrogel. And what this does is this creates the type of exponential concentration gradient that we really need, because in the smallest part of the triangle, we'll have a quicker diffusion, and then on the longest part, we'll have obviously a slower one, generating this gradient that we talked about, and then also having two pools, one above and one below, feeding the organoid with Wnt and Sonic the Hedgehog signaling molecules. And then just another picture so you can see it a bit better and sort of get a gist of the dimensions. And yeah, so we 3D printed our prototype with resin. So obviously this is just the first step. And as you can see, we have here the culture chamber, which is filled with hydrogel. And then we also have hydrogel in this triangle-like shapes. And yeah, just so you can see it. Obviously, this is resin, and we want to migrate into PDSM, which would be the final material. But we're still working on a couple of details, which you'll see a bit later. So we figured we'll try with resin first. So we tried to add the flow. So we have just water with food coloring to sort of try to mimic how these gradient works. And we immediately ran into some troubles. So we have a bit of leakage and air pockets, but this is just the first prototype, and we're actually working on fixing those things So we can move on to the next level, which would be actually cultivating the cells in it and see the way they behave. And then finally, also to understand the diffusion rate of the biomolecules across the hydrogel. So obviously, so depending on the size of these molecules and the diffusion coefficient will have a different behavior across the hydrogel. So we try to model this using the equation that we talked about earlier to sort of understand that and try to fix our microchannels. So we would have the diffusion of all the molecules for it to be constant and for them to happen at the same time. And then finally, I just want to talk a little bit about regenerative design. So we use fusion for this, where we fed the AI our first prototype of the microchannels with the triangle structures. And then what the AI does is it optimizes our design so we can have a better flow of our cell culture medium. And right now, fusion only works with liquids, so mechanics related to liquid and flow. But since it's an algorithm, we can manipulate it and then add our own information. So we can perhaps model protein diffusion or cell-protein interactions, cell-cell interactions, and then have an AI which optimizes our organoid design. So yeah, this is just the optimized design that the AI gave us as an output. As you can see, the input is a bit thicker, so the microchannels are thicker. And then also it's changed a little bit the shape of the triangle. And it just gave it like a more organic shape overall. And then finally, we simulated the cell culture medium's behavior across these new microchannels. And then finally, just as a perspective outlook, so the idea of building this model is obviously to understand neurulation in humans, but also to try to model diseases related to the neural tube. I'm specifically interested in spina bifida and myelomeningocele and defects of the spinal cord. And then we also want to achieve some sort of three-dimensional visualization using light sheet microscopy to understand where each of the cell populations are located, and then finally have single cell transcriptomes with a RNA velocity to infer gene regulatory networks on the pseudotime as well as cell trajectory. So thank you so much for your time. If you have any questions, I'll be happy to answer them. And if you want to reach us, you can. Thank you.
¿Tenemos alguna pregunta del público? Excelente.
Thank you. Can you expand on the use of this tool for the artificial intelligence modeling, let's say, of the channels? I was not aware of it. It's very interesting.
¿Este es el micro? ¿Me escuchan? ¿Qué? Perdón, no había entendido. Yeah, so we used Fusion, which is a software that helps us model and design three-dimensional structures. It's actually used for liquid mechanics, so to build different prototypes for more mechanical aspect of things. But I figured I wanted to give it a try with organoids. And so what this prototype or AI does is that you feed it information about your flow. So we'll have the properties, in this case, of our liquid, which is our culture medium. So it'll talk about the velocity of the flow, the viscosity, the pressure points, if you want it to be constant, if you want it to change. So it'll ask you a bunch of these questions, and then it'll try to use AI to optimize your design so that depending on what's important to you, it'll sort of help that. So for example, if you're interested in having constant flow and have like no change in pressure, it'll change the structure of your microchannels in this case to allow for a homologous pressure alongside it. And right now this only works for liquids, but since it's an AI and it's built on an algorithm, it's actually in Python, so we can add new algorithms to it. So you, for example, in my case, where I want to model diffusion of proteins, what we can do is we can add the diffusion coefficients and then all these different equations that describe the diffusion of proteins across liquid and then across different materials such as the hydrogel, and then add that to the AI algorithm, and it'll try to add, so we have this model of our flow, and then it'll add particles to it and have the particles, or model how the particles move across the actual liquid and then the hydrogel. And then obviously this is something way in the future, but we could also use it to model how cells interact. So what's so interesting about it is that you can add more and more information. So it's very versatile.
Thank you. I think your model is very interesting to make maybe some vessels besides a flow, maybe some vessels into this kind of system of model, but well, maybe it will be later. But actually my question is about in the long term, how much can you maintain the neural tube organoid? And these organoids are allowed to form like more complex structures, maybe cortex, maybe interaction with microglia or oligodendrocytes maturing inside.
Sure. So yeah, so most of the neural tube models usually last about 4 days. So we do have that complicated because actually neuralization is a rather quick process. You know, it usually takes a couple of days. So yeah, so we are thinking about sort of like this 4-day process, and then if we want to continue neuralization into exactly what you're talking about, so have these neural progenitors try to make them more specific, their cell fate towards maybe the parts of the brain or the spinal cord, and have like actual mature neurons. We can do that. I personally haven't really seen it reported in an organoid model, but it's definitely something that we could do. And the fun thing about this type of models is that they're very versatile and you can really push it as much as you want and as far as you want. So it's definitely something that we could do. So we would just have to change the general structure, because obviously there's different types of signaling molecules and patterns that form on different types or different phases of the development of the central nervous system, but it's definitely something to keep in mind. Thank you.
Okay, so theoretically you can keep and maintain it to matureness.
Yeah, I mean, so we would have to see how the cells continue, because like, as I said, I haven't really seen a model that they, or at least the neural tube model that they let or allow it for it to grow for longer periods of time. So theoretically, I would think yes, but we would also have to see how it is that these cells start behaving on longer periods of time.
Yeah, maybe changing the chemotactic pattern. But talking about these patterns, how do you have model, or are there any reports on how those different gradients will be entering to your hydrogel? I'm thinking about they like stopping or maybe entering different like different times.
Yeah. Yeah, so we have thought about that a little bit. So as you can see, we modeled the diffusion rates of our proteins or biomolecules across the hydrogel, which measures 20 millimeters. And so as you can see from these images, the diffusion changes. So some will perhaps diffuse at a higher rate than others. And so we do have to contemplate that when considering how these proteins will enter the actual culture chamber. So we're thinking about maybe having some microchannels that are a bit uphill to sort of slow down those proteins or molecules that move a bit faster than others. So it's still very theoretical because we haven't done the actual experiments yet. This is still very new, but it's definitely something we're contemplating. So we're going to try to do this in the actual device once it's perfected, and then try to see what other things we can change so that the diffusion of all four signaling molecules is constant and that they enter at the same time.
Thank you so much.
Thank you.
Any other question? No? Okay, thank you very much.
Thank you.
Okay. Bueno, con esta plática terminamos nuestro primer bloque del día. Tenemos tiempo justo para hacer un lunch y regresamos aquí a las 2 de la tarde para continuar con el segundo bloque de ponencias del día.
application, and I will try to give you a flair of that also. So briefly, since I'm a bit introducing the topic, about immune cell receptors, you have seen in the presentation of Jay Grove the TCR PMHC complex, so I will not spend too long on that. But here they are represented, so here at the bottom you should imagine that there is a T cell, so the T lymphocyte making a physical contact with another cell at the top, which will be the antigen presenting cell. And then a very specific molecular bond contact is made between the T cell receptor and this peptide MHC complex carried by the antigen presenting cell. And the size about this intercellular distance is of the order of around 10 nanometers. So I will talk a little bit of that at the beginning of my talk, and you heard a lot from Jay Grove. But I will talk more about antibodies. And antibodies, actually, they are initially receptors at the surface of B lymphocyte, so another type of lymphocyte. And at this surface, they are maturating. And once they have a sufficient affinity for their antigen, they can be released in the blood circulation and in the whole body. And they will have the possibility to recognize their target as a soluble molecule. So we'll see a lot also a bit in this presentation, the role of this type of receptor, which is called Fc receptor. And this receptor, they have the capacity to bind this part of the antibody. So an antibody recognizes the antigen through these two FAB fragments. And this constant fragment can be recognized by specific receptors from other immune cells, in particular natural killer cells and macrophages. And so here it's another generic example, but here it's, let's say, an integrin, some adhesion molecule, which is also playing an important role with immune cell, but which is not specific to them, of course, whereas all these three types of receptors are really specific of immune cell. But integrin, they are playing an important role into the adhesion and signaling of this immune cell, and they can adopt different conformation. So a brief introduction about one aspect of immunity, and in particular the role of antibodies in immunotherapy. As I said, the antibodies, they are displayed initially as receptors at the surface of B cell. And there is a selection mechanisms where some antigens here at the bottom are presented by an antigen presenting cell, typically some dendritic cell. And the B cell will try to, there will be contact between the B cell receptor and the antigen. And the B cell will try to pull on this and to see whether it can successfully include, like, integrate the complex antibody, or let's say B cell receptor plus antigen. And if successful, it means that in fact, the B cell receptor presented as a certain affinity for the antigen, and this will lead to some signaling and proliferation of the B cell. And this is through an iteration process like that very high affinity antibodies will be finally produced by B cell after a cycle of selection and mutation to make really affine antibodies. As I said, once they are maturated like that, they are released in the blood, and then they can serve as bridge, typically in one of their role. So here you have antibodies which recognize their antigen. So let's say a tumorous antigen on a cancer cell, here in red, and by this Fc fragment and this Fc receptor that I presented just before, here at the surface of some natural killer cells, so a specific immune cell, which is, as its name, dedicated to kill other cells. Then a molecular bridge will be formed between these two cells and lead to the activation and then to the killing function of the NK cell by degranulation towards towards killing the cancer cell. And so in both processes, it's now more and more accepted that the mechanical force are actually playing a role in these two contexts. Actually, I should have shown that this is the magnification of a conventional antibody, which is formed by 12 immunoglobulin domain. So these are the FAB fragments, the FC fragment, and this is typically 16 nanometer is the size of a conventional antibody. So with a close collaborator, Patrick Cham from the Cancer Institute in Marseille, we specialize in using some single domain antibody coming from camelidae, like this llama, but also camels, for example. And they have normal antibodies, but they have also a specific class of antibody from which one can extract this single domain antibody. So they correspond to basically one of these 12 domains. But in fact, whereas on conventional antibody, the recognition is performed of the antigen is performed using two of these domains or two of these domains here. Here, the same recognition is performed via only one domain. And this is quite advantageous because you have a more stable and small molecules which can serve as bricks to make more complex ones. So what the team of Patrick designed, these are bispecific molecules which will have here, so 4 domains and 2 specificities, one, let's say, for the antigen, this one, and then one for the immune receptors. So for example, the receptor of the NK cell, so that this molecule can serve as a bridge here between these 2 cells and replace the conventional antibody. This can be interesting because in certain people, you have some mutation of this Fc receptor, which make them quite inefficient to recognize therapeutic antibody. So for example, for breast cancer that we are interested in, a very standard treatment is to use a conventional antibody, which commercial name is Herceptin. And it's largely working through the efficiency to treat cancer of this molecule is essentially or largely working with this mechanism. But for certain patients, the recognition between the Fc receptor and the Fc fragment of the molecule is not working, and so these treatments are not working for them. So if instead you use an antibody which bypass this mutation, then you can have efficient molecule even for these patients. So this was a bit the bio introduction, but since I'm a physicist, I will give a bit a different perspective now to introduce the presentation, which is the binding of ligand and receptors in 2D and 3D. So typically, if you are interested in molecular interaction, you can think of a soluble ligand binding at the surface of an immune cell. This could be, for example, a cytokine. I told you that the immune cells, they work with diffusible agents. So that can be one of the situations where you have this kind of binding. And in this case, you have a so-called 3D interaction because the ligand is diffusing in solution before binding to the surface receptor. And you have only one bond to consider, where usually the affinity so the chemical affinity is a parameter of sufficient interest to qualify the interaction, even if you could go a bit more advanced into characterizing the kinetic of the reaction. But at least you have only one bond here, and the affinity measured in solution is the right parameter. The situation is quite different when you have a cell-cell contact, and in particular the TCR PMHC, and this was again the topic of Jay Grove's presentation. In this case, you have so a 2D interaction because both the T-cell receptor and the PMHC are confined to the surface of their respective cells. Their motion is restricted, and you could not anymore apply the same affinity constant that you would have measured in solution because the motion of the molecule is actually modified by their presence at the surface. So this is a so-called 2D interaction. And in this case, from the physical point of view, you can imagine that the distance between the two membranes, so for example, if the two cells are too far apart, for sure the two molecules will have no chance to bind. So you have to take into account that to describe this interaction. And if also the two cells have, are subjected to motion and so on, then this bond will be subject to a disrupting force, and therefore could be influenced by this force, which of course is not the case when one of the ligands is, or one of the partners is soluble. Now, the third situation on which I will mostly focus is the one where you use an antibody as an intermediate bridge, because here now you have a 2D interaction. Still, the main receptors are on the surface of two cells, but in between you have a third partner, which is the bridging molecule. So here you have two bonds to consider, actually. The bond here between the Fc receptor on the immune cell and the antibody on the Fc fragment, or the bond between the antigen, for example, in the case of the breast cancer, will be HER2, for example, and the FAB of the antibody. But since you are in 2D, you have also potentially the effect of force and distance. So the system is actually much more complicated in this case. So to study that from an experimental point of view, you can go from cell-cell interaction also to some reductionist approach. And this is typically the system presented by Jay. So you can replace one of the partner by a surrogate partner cell. And so, of course, in the case of a molecule, you will just, let's say, study the bond between ligand and receptor on a glass surface. I will show you some example. And here, this is the system which was described by Jay, where you would have, let's say, a lipid bilayer, or you could even anchor directly the ligand of the immune receptor on a glass surface and study with good detail the immune cell behavior. What I will show you is that we can also adopt this strategy, but having an intermediate molecule as a molecular bridge. So as I said, in this configuration, not only you can tune well the surface density and the organization of the molecule, as Jay was showing, as it will be presented by Keia tomorrow, but also you can do high-resolution microscopy in this configuration. So as an outline, I will tell you about this situation where we can measure in vitro only with molecules, so without cells, the molecular unbinding, especially when we apply force on the molecule, on the bond, how it could be correlated with cytokine release. And then in the second part of the talk, I will talk about cell spreading mediated by an antibody, or in the last part of the talk, about antibody-dependent cell cytotoxicity mediated, so again, by different type of antibodies. So in the part one, I focus on this and start with the biophysical technique to measure the behavior of bond, so ligand receptor bond under force. And we got an example given by Alex Dunn in the second presentation of this seminar on Tuesday, where he was using magnetic tweezer. So I will show you briefly about that. But before, I would like to introduce a method which was actually invented in our lab. But before that, Pierre-Henri and me joined by Pierre Bongrand, the founder of the Laboratoire Adhésion and Inflammation, which is called the laminar flow chamber, and where so you will put, let's say, some ligands on a glass surface, and you will have use microbeads coated with the corresponding receptor. And all this will be carried and pushed by a laminar flow in a fluidic circuit and observed in an inverted microscope, in bright field microscopy. So relatively simple setup, but which is actually pretty powerful. And the way it works is that, so the beads, they will be dragged by the flow. So you see here the image of beads going to the left, just pushed by the flow. If they are approaching the surface, they will have a chance to make a molecular bond between the surface of the bead and the bottom, because they have corresponding receptor. And if I play briefly the movie again, you may notice that some of the cells will arrest sometimes, like this one, you see. And sometimes they restart, not all the time, but basically in this condition, we make sure by diluting the molecule at the surface that once a bead is stopping, it's because one molecular bond has been formed. So you can directly identify when a bond is formed. And if the bead is released, it's because this same bond has been broken. So by measuring the duration of the arrest of the bead, you can directly have a distribution of the lifetime of the molecular bonds. So, and this, you can do it at different flow rates, where you will impose each time a different force on the bond. So you can actually retrieve the dependence of the lifetime of the bond as a function of the rupturing force. So that's what we did with Philippe Robert, a long-term collaborator in LAE, who worked a lot on the flow chamber. And we have been interested in studying the TCR PMHC bond under force. And at that time, there was some suggestion or even hypothesis that in fact, the TCR PMHC bond, in order to produce the activation of the T lymphocyte, was requesting that it's a catch bond. So you heard about the catch bond in the Alex Dunn presentation. This is a molecular bond, which here, if you look at the the half-life, let's say, or lifetime of the bond, that as function of the force, it will behave like that, that for low force, it will be low, then increase to a maximum and typically decrease. So it's actually catch-slip bond, but basically, you have a force at which you have a maximum lifetime. So people at that time were convinced, or many of them were thinking that you need such a bond to have a correct TCR, a correct T lymphocyte activation. So what we did here in this work is that we took different peptides. So remember, TCR is recognizing peptide MHC. These 5 peptides here, they have the capacity to activate T cells. And so we measured the dependence of half time as function of force for each of them in the flow chamber. And we saw that, as you can see, their characteristic is rather overall resembling the slip bond, and there is no catch bond in this system. So it means that, at least for these 5 examples, activating peptide, they don't need to form a catch bond with the TCR in order to activate the cell. And to go a bit further, we were interested in correlating this off rate for different force as a function of the gamma interferon production, which is a readout also of the activation of T cells. So here you have in the x-axis this gamma interferon production, and you can, for each of the force we had applied, so from 6, 10, 15, 30, 45 piconewton. Actually, here there is even the zero force case. So if you look at the zero force case, which is measured with a different technique here, with a surface placement resonance, you see that the correlation between the off rate and the interferon gamma production is quite weak. So basically, the off rate is not a good predictor of the activating capacity. But now, if you focus on these guys here, you see that the correlation at low applied force is much better, and it starts to deteriorate at higher force. So here we are showing that indeed, at a certain level of pulling force, then the off rate that is modified by the force correlates quite well with the activation signal of the cell. So now I will turn just to a more, let's say, technological advance that we did recently. So I told you about magnetic tweezers. So you heard about it by Alex Dunn. So here I show you different methods for single molecule force spectroscopy using field. So here the field is a laminar flow. You can use a magnetic field to actuate a bead. And you can also use a pressure field. It's the case of the acoustic force spectroscopy, which is the most recent method. And we applied this method actually with an original design from collaborators in Paris, which have designed a molecular scaffold. So this red here is a DNA strand, double DNA strand. And this DNA strand, it can be bound on the lower surface here, so at the bottom, or at the bead surface at the top. And in between, you are interested in measuring the interaction between ligand and receptor. You can graft on this scaffold the protein at two different places like that. And I will show you why is it a very useful system to study bond rupture into one of these two setups. So these collaborators, they had demonstrated this system with the magnetic tweezers, and we have also, I will show you that we applied it also for the first time to the acoustic force spectroscopy system. So the experiment goes like that you have a temperature control microfluidic chamber that you can put on top of an optical an objective, a microscopy objective. And then on top of this chamber, you have a transparent piezoelectric. And you can, by vibrating this piezo, create a pressure gradient within, so in the vertical direction within the chamber. And what will happen is that here you have node of pressure in these two places. So like the beads, which are, let's say, at the bottom of the surface, they will be attracted towards this node pressure. What will happen for the scaffold is like that, is that before you apply any pressure, pressure or force, basically, then you know the scaffold will be released, and then the proteins, ligand and receptor, have the possibility to make a bond. Upon at a certain point, you will decide to apply the acoustic force, so it will pull on the bead, and the scaffold will extend and put this bond under tension. And if you follow the vertical position of the bead as a function of time, you can, by measuring this height, make sure that the scaffold is in this configuration, so where the bond is attached. Okay, so and so then you just record, and at a certain point, the bond may be breaking. This is what happened here. And at that time, the scaffold will extend. But here, the trick is that there is a leash keeping together, let's say, not preventing the ligand to completely escape. So we just record a difference in height between the closed and open state due to the size, the length of the leash. Okay. So here, in fact, this is the open configuration, which is recorded here. Now, so here at the end, here the time that you record, this is the time of the bond, which was under the exerted force. Okay, once you are here, you can release simply the acoustic pressure and go back to the initial state where maybe you will release the force, the bead will just diffuse again, and then you can start that again. In practice, you can repeat this cycle as many times as you like, more or less. Here it's typically 100 times, okay? And each time you see the red trace, it's because it's the closed state. And in each of these closed states, that when you zoom in, you will see that, you can measure this duration, which is the lifetime of the bond under a certain force, which depends on the applied pressure. So here, I plotted the survival curves, meaning that what is the time the bond is surviving as function, I mean, how long it's surviving. So here at 0, all the bonds are formed, and then over time they will break apart as time is passing. And these different curves are obtained for different applied pressure, which correspond to different force. Now we need to calibrate this. So I will not go into detail, but we had to make quite some effort to actually know the exact force which was applied on the bead in this system. So I will not go into this detail, but once we manage that, we can make this kind of plot where we will measure the off rates, which basically correspond to the slope of these survival curves. So this is the off rate for different applied force. Here we are in a range between 0 and 15 piconewton. And you see that the off rate is first as a certain value at 0 force. We call it the off rate at 0 force. This is what you could measure in solution in 3D. And you see that the off rate is increasing with force. So it means it's a slip band. While you pull on it, the off rate is larger, meaning that the lifetime is shorter. And here, interestingly, each color corresponds to a different molecular bond. So we can really establish these statistics. Here, there is a set of maybe 15 or 20 different bonds. And for each of them, we can establish this curve and plot the off rate at 0 force as function of the well barrier distance. So this is what is characterizing here the slope of this curve, right? This basically means what is the sensitivity of the bond to the applied force. So 2 remarks is that, so in here you have other measurements made either with the magnetic tweezers or for the red ones made with some ensemble method where you just determine the off rate at 0 force. So we are not very far, but interestingly, you see that there is a certain variability, basically that different molecules or different pairs of molecules behave kind of differently in a mechanical manner. Okay, so I come to the second part of my presentation, which will be more at the cellular scale. And as physicists, we were interested in doing our own experiments, spreading or ADCC, but we realized that the analysis of our microscopy data was quite difficult. And with a talented PhD student who was co-advised by Keia and myself, who defended in spring, he developed a full software called Cell Detective, which can work with 2D stack images, so time-lapse stack image with multi-channel data, and typically to work on samples that immunologists are often using with multi-well to vary condition, biological condition, and study, for example, the effect of different concentration of antibody in antibody-dependent cell cytotoxicity assay. So this is the kind of data we have, and this is what the software can do. You can pre-process the data, and then it's made to analyze two type of cells simultaneously. So here we applied it mainly to the co-culture of effector cells, the immune cells, and the target cells. And so you can perform a series of segmentation, tracking, and measurement on each of these two populations in order to retrieve time series data. I will show you why it's interesting to do that. And once you have done that, you can also look one step back in your data, because you can then look at the environment of each cell and see whether the local environment in terms of target or effector is playing a role into how the cell is behaving. And we can do that even in a more time-dependent way. I will not talk too much about this last application. So this software is in Python. It's freely available on GitHub. And the data, demonstration data, are on the Zenodo repository. So here is briefly how it looks like. This is the main, so you maybe don't see much the detail, but this is the main working panel where you can select pre-processing, process effector cell, or target cell. And when you process effector cell, you can perform the segmentation either in a traditional way or using AI methods, including StarDist or cell pose method. You can make correction. You can even use this correction to retrain a model by re-injecting this data into a new model. You can do a trajectory, follow the cells by tracking using a Bayesian tracker. And you can do, of course, single cell measurement. In all these cases, you have also the capacity to check out the results directly into the software by harnessing, in particular, the Napari plugin. So a first application is a cell spreading assay. So here is how the experiment looks like. You just drop some immune cell on the surface which has antigen. And as I told you, some antibody that we are interested in studying. So this is how the sequence look like. These are data from Dahlia. And we observe these cells with our favorite microscopy method, which is RICM. It was mentioned yesterday in the talk of Daniel Rivlin. So it's a very powerful way to measure the interaction zone between cells and a surface. And you don't need any label, any fluorescent label in this case. So it's very convenient because here we can use primary human natural killer cells without disturbing them, and still so no labeling of any kind. We can still very well study their interaction with this surface. So here on the surface we have the antigen R2, which is the so-called cancerous antigen. We can control its density, and we have inside also the solution here one typical bispecific antibody for which we can control the moiety which recognize the antigen and the moiety which recognize the Fc receptor. So we use cell detective to study and to analyze this data, and this is what the segmentation is giving. So in orange you have cells which have spread and which can be well separated. In blue you have cells which didn't spread. So here there is a decision. Some of the cells decide to spread and some not. And one important question is to understand why and how does it work. And you see that there are also other cells which were not segmented because simply they are not NK cells, they are red blood cells which were remaining due to a limited purification from the blood. So you can look into detail after tracking the cell into the intensity versus time. And this is what happened when a cell, before a cell is landing. If you look at the intensity, this is the background. But once it's approaching the surface, then you see a rise in intensity. It's slightly whiter during a certain time. And at this time, so we can already here notice that we have a contact time that we can mark here at this point. And during a certain time, the cell is overing. And at the point, it decides to start spreading. So then the signal becomes darker. So the intensity is dropping below the background. And we can here mark a second time, which is the spreading time, and measure the overing duration. So how long the cell takes to decide to spread. So this is what it gives on a large number of cells. In blue, you have the cells which decide not to spread, and they have been aligned with the contact time. So here, this is the contact time. I mean, they are all aligned with that once this time has been measured for each cell. And on the right side, so here this is the intensity, and here this is the area, which of course is close to zero and not significant when they don't spread. And for the case of the cell which spread, then they have been aligned with the spreading time. And see here, you can see that there is a nice global behavior of intensity as function of time for all the cells which decide to spread. And at the same time, we can measure the area as function of time for these cells, and we see that it's increasing, but you still have quite a variation of the final area of these cells. Here you have maybe about 100 traces which have been automatically analyzed by the software. So with this data, you can, as I said, you can measure the overing duration, and you can plot against survival curve. This time, how long the cells are overing before deciding to spread. So what is the fraction of them which didn't spread for a given time? If you use a very low concentration of antibody in blue, then most of the cells don't spread. They decide not to spread. But then slowly, once you increase the concentration, then you have a larger and larger fraction of cells which spread, which with a kind of characteristic time. So here, if you take the slope of this survival curve, this is indicating the velocity at which, or if you like, the rate at which the cell decide to spread. And this is increasing with the amount of antibody. And of course, this is really a question on how this mechanism is working, and this is a subject of future investigation for us. Now, you can also look at how the cells are spreading. So in particular, you can measure the spreading rate as function of antibody concentration. So at low antibody concentration, not much data to study because few cells are spreading, but at higher concentration, then you can start to measure this velocity for a large number of cells. You see that you have a large variability in the spreading rate for intermediate antibody concentration, whereas it starts to saturate with a bit less of variability for higher concentration. And here also, it's interesting to try to understand what happens here. I will now talk about the last part of the presentation, which is about antibody-dependent cell cytotoxicity. So this is actually an experiment which has been set up in Marseille by Beatrice, a Mexican postdoc who was working previously in Lance Biodit. And so she set up this system where you have a co-culture of target cells with effector cells, so these primary NK cells, for example, in the presence of the antibody molecule we are interested in testing to see where their efficiency in helping killing the target cells. So you put all this on a microscope with controlled temperature, ideally controlled CO2, and you record some movies, which will look like that. You will see a magnification of that after. And previously, you have, Beatrice, in this case, have labeled the cells with fluorescent markers, in blue for the nucleus of the target cells, in green for the effector cells, and you have also propydium iodide, which is a death marker, which will show up as red once a dead cell, once the target cell is dying. So here at the beginning, all the target cells nuclei are blue, and then over time you will see that more and more are becoming red. It's about 2 hours here of recording, and at the same time you can see also the behavior of the green cells, which are the natural killer cells. So Cell Detective software is helping us to analyze this, and I will skip the details, but this was quite a challenge to analyze, and in particular to start segmenting this. And the usual AI software didn't work directly, so we had to tune them because you have a different cell population of different size. You see that some of the labels are also going, for example, slowly the green cell will become partly blue. Because they will integrate some of the UXT. So this is complicating the matter. But ultimately, we managed, and mostly Rémi actually managed to do that. And this is after tracking of the target cell, what you obtain in the same way, same type of presentation as the previous application. We can distinguish the cells which don't die during the experiment with the cells which die. And here we follow the intensity of the propydium iodide, so the death marker. So here, of course, for the non-dying cell, it's remaining at a very basal level. But for the dying cells, you have a sudden increase of the PI level at the time of death. So here, all the dying cells have been aligned with the death time T0, which was automatically detected. And you see they have a quite reproducible intensity response in terms of the propydium iodide. Interestingly, you can look at any other parameter that we measure here in the system, for example, the nucleus area. So on the non-dying cell, nothing much happened. Their area is quite constant, even if you have, of course, a variability between cells. But if you look at the dying cells, you see that at the time of death, you have a very neat decrease for all of the cells, so that at the point of cells is dying, at least as indicated by this propydium iodide, the nucleus is actually retracting, and we attribute that to the fact that at that time the cell starts to deadhere and release a pressure on the nucleus, which makes it look more like a round shape and decrease its apparent radius. So here it was interesting to follow finally another marker of death, which could be taken completely independently as this one to measure this test time. So you can use this data in an ensemble way to draw survival curves, so the fraction of cells which survive as function of time for different antibody concentration. So this type of data you can obtain with an ensemble method, but here we can do more. We can say, okay, now for all the cells who die or don't die, I will try to see what is around them. So for example, do they have few target neighbors, or do they have a lot of target neighbors? So you can split arbitrarily the population into two. For example, this one, which has a few target neighbors, let's say less than 3.5, and you can draw the survival curve for this subpopulation. And you can do it also for the other subpopulation, which have a larger number of target cells, and you see that there is a clear difference, meaning that the cells which have a higher number of neighbors, they are actually surviving better to the killing by the NK cell. And so this is interesting to study the reason for that. So we are going on studying that, why this is the case. So this is the kind of application we can perform with this assay and this type of analysis. So we can do that also with the natural killer neighbors. So we can ask the question, we can count around the target cell how many natural killers are there. Okay, and we can now make some subpopulation of target cells which will have different number of neighbors. And for each of these subpopulation, we can draw some survival curve, and we can measure the lysis rate. So the slope of this survival curve here as function of the number of the typical number of NK neighbors in the neighborhood of each of the target cell in this curve, containing this curve. So here, you have a control without antibody. So you have a fairly low lysis rate, but which is non-zero because the natural killer cell, they kill anyway, even if you have no antibody in solution, but at a slow rate. If you put 1 nanomolar of this specific antibody, then you have a strong increase, of course, in the killing rate. And very interestingly, you have a linear dependence between this of the lysis rate as function of the number of NK, so the number of NK neighbors. And it's interesting also to try to interpret this type of linear dependence. We suspect that this is a signature of the fact that only a small fraction of the NK cells are capable of killing in this case. But we are going on analyzing this data to try to prove or disprove this hypothesis. So to finish this presentation, I will show you something a bit more on the modeling side, but still on this tripartite system. So NK cell, B-specific molecule, and target cell. And here, a relatively practical question is to say, okay, how much antibody, how much B-specific antibody do I need to kill, let's say, for example, 50% of the target cells? One understands that the more antibody you put in the solution, the more will be the amount of killing of killed cells. But how does it behave quantitatively? And to our big surprise, there was absolutely no insight about this in the literature, even if these specific molecules are more and more developed for therapeutic applications. So this is the problem that we pose. And in our case, we can construct different B-specific molecules by assembling different bricks. So I told you, for example, we have 2 single domain antibodies which can recognize the effector site, so the Fc receptor on the NK cell side. And we have here 3 single domain antibodies which can recognize the antigen on the target side, which is here R2. So we can make 2 times 3, 6 combination and 6 different antibodies based on these 5 bricks. Okay, so here I show you the data which were mostly compiled by a Spanish PhD student, Cristina Gonzalez. So she made many experiments, a bit simpler than what I showed you before. Here we just measure the endpoint killing. So we just mix the two cell population, the target. So we have 3 target cell line. We, over the time, so each time we could, at each experiment, we would get NKs from a different donor, so from different LD donors. So here, together with another engineer in the lab, she accumulated data from 15 donors. And all this for our 6 bispecific antibody. You mix one of each of these 3 together, you wait overnight, and then you measure what is the fraction of dead targets. And this is what it gives. So it's a bit fuzzy, but there are a lot of data. So here you have 3 columns corresponding to 3 target cell lines. So here this is a classical low expression of HER2. So it's supposed to be a cell line MCF7, which is kind of negative for as model in breast cancer. Usually in breast cancer, the cells, they are overexpressing HER2, and this corresponds more to the case of this SKBR3. Here, in addition, we made a new cell line, which was derived from the FCF7 wild type, to overexpress HER2 to a similar level as the HER2 positive cell. And so for each of the 6 bispecific antibody, we draw the lysis fraction after 1 night of co-incubation as function of antibody concentration. You see that the response is always like a Hill function. I will show you the detail of this function. We can fit each of these data, so there are around 200 conditions here, with a Hill function. And this is so in practice, if you take a single condition, you have the lysis fraction as function of concentration, and you can fit with 3 parameters. The minimum value, so at 0 concentration, is correspond to the spontaneous kill fraction without antibody. The maximal one, and then the concentration EC50, which is called the potency, is basically the concentration of antibody, which gives you half of the cell killed. Okay, so you can fit with a simple equation. And when you dimension the data for all of the 200 conditions using mean and max or EC50, they all match pretty well on this Hill function curve with a fairly limited residues. So here we have all the data from 200 conditions, which represent approximately 2,000 points. So here is, so all the data are summarized, most of them are summarized actually in the potency. That's what we were interested in. And, but you can plot this potency for the different antibodies, okay? So we have 6 antibodies, and you see that, interestingly, some requires much less antibody, much less concentration than some other ones. So they don't have the same potency. Another interesting remark is that if you look at the scale here, we are in the picomolar range. So you need very little of these antibodies to get half of the target cell killed. And the third observation is that here each color corresponds to one of the 3 target cell lines. And maybe as expected, if you consider the blue one, which is the low density of antigen, MCF7, of course, you need more antibody to achieve the same killing. So the EC50 is higher at the low density, whereas the 2 other at similar densities of HER2, they behave quite similarly. Now you can also decide to rank the data in terms of the donor, because this is also a factor of variability. And indeed, if you look here, they have been ranked according to their to the potency on the high density. So, for example, the orange one, and you see that you have also a significant variation in the EC50 depending on donor. So all donors don't react similarly. You don't need the same amount of molecule to achieve the same result depending on the donor. And you get also different values on the MCF7. So we wanted to understand that more quantitatively as a physicist. And so we decided to design a multi-scale model. And there are 3 ingredients in this model at 3 different scales. That first, we need to understand how the bridging by the B-specific molecule is occurring at the immune synapse, so at the contact between the 2 cells. Then we make an hypothesis on how this bridging density will affect the killing rate. And then we need to compute that for what is our measuring quantity at the population scale. So first, how the B-specific is binding at the interface. So here we make an hypothesis of equilibrium, which is a simplification. But you have to understand that in order to make a bridge between the HER2 and the CD16, so the receptor on the immune side, you have 2 ways to do that. You can bind first on the left in the step A, and then you can bind on the right in step B. Or you can first bind on the right during step A, and then further bind on the left at step B. Okay? But if you are at equilibrium, there is a principle which is called the detail balance, which imposes that you can run this cycle. And this imposes that you have a relation between these quantities and these quantities. And indeed, so for example, the reaction which happens here between the green molecules in step A, this is the same reaction, at least this is the reaction between the same partners which occurs in step B. Okay? So there is a relation with a factor between these 2 quantities. And because of detail balance, the same factor alpha that we call cooperativity is intervening to characterize this affinity constant. So here we are at equilibrium. We simplify by considering only affinity constant to describe this. So we can easily compute this bridging density. It's a quadratic equation in the, so this is the density of bridge, okay? And this depends on different factors. Some we can measure, the affinity constant, K1, K2 in solution, the density on the 2 surfaces of each partner cell, sigma 1, sigma 2, and 2 parameters, the alpha, the cooperativity, and another that I will introduce here, only the cooperativity is an unknown parameter. So if you draw sigma as function of C, you will see typically this kind of bell shape. Okay, so that if you put too much molecule, it becomes, in fact, it becomes counterproductive because you will fill each of the 2 sides and prevent the molecules to make a bridge. That's why it's going down at high concentration. So if you think I put a lot of the molecule, it will work better, it's wrong. It will work not good because you are preventing the bridging. Now I will, we make a simple hypothesis on the lysis rate, and what is important is how the lysis rate, so the speed at which the lysis occurs depends on this bridge density. So here you have 2 terms in the lysis rate. You have a spontaneous lysis and you have an antibody-dependent lysis, which varies as a function of the density of bridge. And by independent measurement, we can more or less, at least qualitatively, justify this form of the equation. Okay, so these are the parameters we can measure independently, and these are the parameters that we will fit in our model. So this response threshold in terms of density and the cooperativity. And this is the output of the fit. First, you see that all the data, this is fitting with the model as well as the previous fit independently, but here we have a much lower number of parameters. Basically, these are typically all the parameters we have. There are still many, but they each characterize different molecules or different donors, okay? So you have the cooperativity characterizing the antibody, and you have the threshold response characterizing the donor, okay? I will not comment too much on these variations, but I will show you a final slide. In fact, this system can be simplified, at least the quadratic equation can be linearized when you realize that the working concentration, I say that we need a picomolar concentration of antibody to work, whereas the affinities K1 and K2, they are more in the nanomolar range. So if you apply this, approximation, you come up with a very, very simple equation of the density of bridge as function of the parameters. And from that, you can deduce the C50, so the concentration giving half of the depth in the model as function of the known parameters and the fitted one, the cooperativity and the response threshold. And when you plot this simple quantity as function of the measure at C50, you get a quite good fit on 4 orders of magnitude, which says that this quantity, quite simple to calculate if you know alpha and sigma 50. And in this, so it's a good, approximation of the measure DC50, and you can decompose this into 3 contributions, one coming from the effector side, with the density on the effector side, the threshold response, one coming from the target side, and one coming from the antibody, which has the 2 affinity constant and the cooperativity parameter. So this is a quite simple formula to make some prediction in this system. And this is what it gives if you just compute, for example, the contribution to the potency of the B-specific and to the of the donor. And you see that here you have even for the 3 different cell lines a quite characteristic value for each of the B-specific. Okay, with that I will conclude and say that for the last part of the talk, I've shown that we have EC50 in the picomolar range. We can predict this value by orthogonal contribution of B-specific and donors. And we identify 2 parameters, one at the molecular scale, the cooperativity, characteristic of the B-specific molecule, and one at the cellular scale, which is the bridging density, which characterizes the NK response. All this is at equilibrium, so it's very simple. I told you at the beginning that surely at the synapse, at this interface, kinetic and force are playing a role. How does it determine these parameters, and how could we refine our model? This is for future work. So to finish, I thank all the students or postdocs which work on that, Rémi, Béatrice, Christina, Dalia, and Adrien. Then the collaborators of LAI, Martine, Philippe, and Pierre-Henri.
Thank you, Lauren. In the piezoelectric force model, how do you modulate the amplitude of the pressure or of the force?
So in the acoustic force system, you basically you have to set the frequency, because you have to work at a resonance frequency, which is determined by the geometry of the chamber and also the liquid you put in, which is usually some aqueous buffer. And then you are basically tuning the amplitude of the piezo movement to tune let's say the pressure gradient. So this is so-called what they call power in this system, and which is basically the power that electrical power that you send into the piezo in order to produce the pressure gradient. And this will be in the low power range proportional to the force that you will apply. But in fact, you have some complicated nonlinear effect, which makes the calibration quite delicate because there is no linear relation between the force and the power you bring into the piezo.
Thank you. ¿Alguien más tiene alguna pregunta? I have a question.
Okay.
Is it possible to evaluate the activation of
signaling pathways during the spreading event of the cells?
Yeah, that's a good question. As you have seen in the presentation of Jay Grove, of course, the signaling is critical in the activation process, and yeah, what he showed the LAT, this LAT condensate. So we didn't really do that yet, and as you could see, Jay didn't look at the spreading, or at least didn't show any much data on the spreading process. But for sure, what is critical is in the interplay between the spreading itself, so the deformation of the cell, and the signaling process. And it occurs very likely, in a relatively coordinated manner. But all these events are going pretty fast, and it's difficult to monitor the two. But one obvious starting point in our case would be to monitor simultaneously with the spreading, for example, to monitor the calcium. But we could also think of harnessing the system presented by Jay to do simultaneously with the RICM, so the spreading readout, to also monitor the formation of the LAT complexes if we have the right cells with the right fluorophores.
Okay.
So that's possible.
Thank you so much. And thank you so much for all of you. Por favor, un aplauso. Chicos, les recordamos que nos sigan en todas nuestras redes sociales de Lanciodit. Ahí pueden encontrar algunos eventos que se van a estar realizando próximamente. También les invitamos a que sigan nuestra transmisión de YouTube de este simposio que estamos realizando. Pueden verlo, las grabaciones se van a quedar ahí de todos los ponentes que han dado su plática hasta el día de hoy. Bueno, para nuestra última plática del día de hoy, me da muchísimo gusto presentarles a mi compañera de laboratorio. Ella es Amairani Ventura. Ella actualmente está haciendo sus estudios de maestría con nosotros en el Laboratorio Nacional y su investigación tiene como objetivo contribuir en el entendimiento de las interacciones entre células del sistema inmune y el cáncer. Ella tiene que trabajar en recrear esos ambientes, en los microambientes que se dan entre las células del sistema inmune y las células del cáncer. Entonces, no me robo más del tiempo de Amairani. Amairani, muchas gracias por estar aquí.
Good afternoon. My name is Samairani, and I'm master student at the Lens Biodit. And today, talk us about the characterization of transmigration and effect of natural killer cell geometry. So first, I start to talk about natural killer cells. These cells are part of an innate immune system, and these cells have a cytotoxic function and are characterized by being the first line of attack against viral infection and tumors. And also, these cells are circulatory cells. A circulating cell, this through different types of tissues and barriers, and understand the migration of natural killer. It's very important to unraveling the importance of natural killer in immune response. But the natural killer not killing all the time. In healthy situation, the natural killer have a equilibrium between activating receptor and inhibitory receptor. But when one cell are infected with a virus of is a cancer cell, the cell lost the MHC class 1, and this is the signal of the natural killer recognized for kill. And the natural killer kill through the release of granzyme perforin granules. But what is the importance to the calcium, to the activities cytotoxic in the natural killer? So in this image, we can see the PIP2 pathway that causes the release of calcium of the reticulum. And this activates STIM1 and ORI1 that allow the entry of calcium to the cell, and this release the granzyme perforin granules. The calcium is very important to the cytotoxic response because its activation of signaling pathway, the granulation and release of cytotoxic granules, interaction cell to cell, cytosine production and immune regulation, and modulation of stress sensor and activating receptors. The importance of natural killer cells is that currently these cells have a lot of importance in the immunotherapy because natural killer cell can be modified with receptors that target tumor antigens derived by lytic function toward transformative cell. But there is insufficient natural killer cell infiltration. So we wonder what is happening with natural killer cell migration, and also how is natural killer calcium signaling when cancer cells take on different morphologies. For this, we designed two microfluidic devices. The first is for evaluate the cell migration in the natural killer. This is the mold. We use a biocompatible polymer that called PDMS. These blocks of PDMS are joined with a collagen membrane. And in this, we put the endothelial cell to mimic a vascular wall. So in one side of the device, we put a cancer cell that we use MCFC7 cell, that is a cell line of cancer bread cell. And the other side, we put a natural killer cell. So microfluidic device make it possible to replicate conditions that we can see in the body. Okay. Then the flow is incorporated in the device, and in this video we can see where the natural killer pass through the microfluidic device. In red, we can see the natural killer, and in green, we can see MCFC7 cell. This is the collagen membrane. Other advantage of this microfluidic device is that this device have the vertical membrane. This allows the observation of migration in a single plane. So we are working with the monitoring of the migration of these cells, and this is an image that the collagen membrane in red natural killer, and these points red are natural killer. So we are working with this. So I need to work with more time of the evaluation of the migration. This is an amplification of this image. For evaluate our second question, we design a second device to control the morphology of cancer cell. So this is the design. In this, we have a substrate glass, and we use PDMS also in this stencil. And in this part, we put the micropatterns of proteins of fibronectin. This is a substrate glass, and the advantage of this model is the precision in cell shape control, ability to stimulate specific microenvironments, and we can monitor in real time of cellular response. This device allows to modify the morphology of MCF7 through the use the micropatterns of proteins. So, what is the importance of the morphology on cancer cells? The morphology of cancer cells have an impact in the calcium intracellular calcium signaling in natural killer, because the morphology in the cancer cells modify the interaction surface, ligand distributions, mechanotransduction and calcium signaling, and the modulation of the microenvironment. Also, the organization of cytoskeleton of MCFC7 have an impact in the calcium signaling in NKCs. For this, we develop a controlled approach using fibronectin micropatterns within a microfluidic device. This is a scheme that the stamp that we use. These have 4 circles with different size, 20, 14, 18, and 120 micrometers. And these are fabricated with PDMS. This is an image that the stamp that we use. And in this, we can put the fibronectin over the substrate glass. This is an image of fibronectin micropatterns on glass substrate for controlling cell morphology. The advantage of micropatterns of protein is a precise control of cell morphology, replicability of the experiment, and high resolution approach to cell interaction. So first, we put the micropatterns of fibronectin in the substrate glass of the microfluidic device. Then we put the MCFC7 cell in the device, and the MCFC7 cell recognized the fibronectin and put it on the stamp. And then the cytoskeleton of my MCFC7 cell are modified, also the morphology. Okay. And then we put the natural killer to see the interaction between two cells. And then we monitor the calcium recording with fluo4. In this video, we can see when the natural killer bright different. We observed that natural killer have a lot of different response of calcium. And then we are working to establish the relationship between the MCFC7 cell morphology and the pattern of calcium. We are working with this project. These are part of my master project and Diana master project. So, thank you. For finish, I would like thank to my multidisciplinary team to work every day to make it possible at Landsbioedit. Thank you.
¿Alguien tiene alguna pregunta?
Thank you. Thank you for your good work. Actually, you have been allowing the interaction between natural killer cells with the MCF7 cells, right? And what is the linker? How they are interacting? Because natural killer cells needs to be activated, like maybe antibody, or because they are sometimes ADCC kind of cells. So how do you see to it that they will have an interaction with the cancer cells?
The MCFC7 cell express a ligand to activate NK2D to recognize the activating receptor of natural killer cell. This receptor is in the natural killer. So.
Okay, and this you are measuring the calcium like signaling, like I mean to say the calcium molecules are being released whenever the interaction happens. So then there is a calcium release taking place,
yes,
right? And that you're measuring using fluor4 cells. Okay, so so you're using basically a confocal microscopy for detection of the calcium which is being secreted out, right? And what is the mechanism of this release?
The release of calcium?
Yes.
The pathway of the calcium?
Yeah.
Okay. The release of calcium is through the P2 pathway. IP3 activate the channels of release calcium on the reticulum, and then this signal activate the STIM1, that is a type of receptor on the membrane of reticulum, and this give a signal of ORI1, that is a type of channel in the membrane of cell that allows to entry of calcium. So this release of calcium cause the migration of granules through the cytoskeleton of natural killer to realize their content in the extracellular space.
Okay, so basically you are, so this is a fundamental mechanism which is existing. So you're proving in your microfluidic system that this is happening in your system as well, right? So that is the idea behind performing this work.
I think that
You're proving this particular process in your microfluidic system. Is that the idea?
I think that
Eso es la idea que usted quiere una evidencia, usted quiere usted quiere mandar una evidencia nada más. Eso es de idea para su trabajo. Eso es mi pregunta. ¿Qué es de idea para su trabajo?
What is my project?
Sí, what is your project? Yeah.
My project is to evaluate the intracellular calcium signaling on natural killer in response to modify of the morphology of MCFC7 cell, because the studies say that the morphology of cancer cell have an impact with the activation of natural killer.
Yes, exactly.
Okay.
That's what I want to, because like there is a morphological change and then that will be leading to the activation of a natural killer cell. You mean to say that, and that's what you want to prove. Okay, and yeah, it's quite fundamental. That's what I wanted to ask you, that exactly what are you interested in? Because you have used only fibronectin. You have not used any other molecules, like apart from fibronectin. Did you use anything else? For micropatterning?
In this moment, we working with fibronectin.
Yeah.
Other projects are working with collagen, but we chose fibronectin because the natural killer have a lot of the MCF7 cell have a lot of receptor of fibronectin, but it's possible to work with different proteins of the extracellular matrix.
Yeah. Okay. Yeah, that's the idea. Thank you. Thank you. Gracias.
¿Alguien más quiere hacer una pregunta?
I had some questions about your pattern.
Okay.
What is the size of the, I think there was no scale bar on the...
We use 4 different sizes of circles, 20, 14, 18, and 120 micrometers. And the size of the space between these circles are 13 micrometers. And the size of the MCF7 cell are, I don't remember exactly, but I remember that is like 13 micrometers.
Okay. And in the same slide, you have different sizes, is that right?
Sorry,
if you show the next slide, if you show the next slide, please. On the same slide, you have 2 different sizes, is this right? Some big circle, some small circle? And the same micropattern?
Okay.
Sometimes, like on the same slide, you have big pattern and small pattern? Or?
Yes, in the same stamp, we have the 4 different sizes of the micropatterns. This.
Why do you do that? Why do you not, have one place with only big patterns and one place with small patterns? Because you want the cells, if I understand right, you want the MCF7 cells to be touching each other, or you want to keep them separate?
Yes, we are working in this, but it's a good idea. And in this moment, we working with this stamp that have the 4 different sites. The idea is to monitor at the same time the cells, but I need to evaluate the possibility to make this, or maybe it's a better idea to evaluate first one and other.
And what do you have outside of fibronectin?
Can
you hear now? Yeah. What do you have outside of the fibronectin? Like outside the green, what is the black area? Do you put BSA or I don't know, PEG or something? Outside the fibronectin.
Outside. No, we wash, and in this moment we don't have a problem to contamination in the substrate glass. Se me fue fuera. The microstamping area.
Okay, thank you.
¿Alguien más?
What is the effect? What do you think is the effect of the flow in your system? Talking about when you put the cells, then they roll over the surface, but the velocity of the flow, do you think have an effect on the migration? And the other thing, do you think that the gravity making the cells going down will affect also the transmigration of the cells?
In the device of membrane?
Yeah.
Okay. The velocity that we use in the microfluidic device of collagen membrane, it's the same that in the vascular wall. So I working with these proofs, but till today we don't have a problem of with this velocity, and we need to evaluate for more time the migration of natural killer to comprehend better the migration in general of natural killer. And what is the second question?
About the gravity making cells going down instead of making them going through the cold one membrane? Because you have the channel like an IU, no? And then the cells are flowing through the channel, and in some point they get in contact with the membrane so they can cross it. So at some point also maybe the cells can go down only because of gravity effect, only because of the maybe sedimentation of the cells.
Well, the flow is constant, so we don't observe the sedimentation on the natural killer cells. And natural killer and MCF7 are cultivated in the microfluidic device, so these are in the substrate of the microfluidic device. We working with this, and we learn about this micro device. So in this moment, we don't observe the sedimentation.
Okay, that's good. That's good. Thank you.
¿Alguien más tiene una pregunta? Maybe it's for Diana more. Actually it's related to Nicolas' question. What strategy do you use to make the endothelial cells you said you cultured to stay on the collagen membrane that I understand that is vertical instead of adhering to glass?
When we culture the endothelial cell, so first in the device, we culture the endothelial cell. So the collagen membrane is vertical between the two PDMS blocks. So we need to put the device in a vertical form. So we put the endothelial cell, and we need to wait 2 or 3 days, and then we put the device form horizontal, and we put in other side natural killer, and the other cell MCF7 cell, are the culture is normal in this moment.
¿Alguien más tiene una pregunta? ¿No? Parece que no. Pues muchas gracias a Mayrani por tu
Thank
plática.
you
Es la primera vez que presentas en un auditorio con un público. Entonces, muchas gracias por tu presentación a Mayrani. Le damos un fuerte aplauso, por favor. Y bueno, solo para finalizar, hoy ya cerramos las ponencias del día de hoy. Mañana los esperamos igual a las 10 de la mañana en nuestro cuarto día de pláticas. Y bueno, ahorita todas las personas que se inscribieron al curso práctica, pues nos tenemos que ir al laboratorio para nuestro tercer día de prácticas. Muchas gracias a todos y los esperamos mañana. No olviden seguirnos en nuestras redes sociales.